Okay, hello everybody. Super happy to see so many of you here. Yeah, Sean has already introduced myself a little bit. So my name is Christian. I'm a research scientist here at the Institute for Systems Biology, and I will be the one guiding you through the course materials today. And we will be talking about Amplicon sequencing data analysis with QIIME 2. And just to kind of introduce a little bit, what will be happening is that we'll have the course, which will be 2 parts today, and that will be followed by a really cool talk today. So yeah, we have a pretty full and super cool program, so I'm excited to go through that. So before we actually start, let's talk a little bit about how this talk is organized. You should have received already a link to the slides in the email, and you can also see it on the Slack channel, but here's also the link if you want to open them now. They're basically a web page, so feel free to open them on any device that kind of has like a white screen, can be your computer, phone, your fridge, if it has a screen, whatever you have available. But that will help you to kind of go along. The slides will also have the links to everything that you will need during the course. So it's like a good starting point to have them somewhere, and feel free to, go back and review slides as you see fit. So the course has kind of like a 3-tiered approach. So there's like 3 parts to it. One is the presentation that you're seeing right now. We'll use that mostly to, give some explanation, some logic, some references, everything that you kind of need to kind of understand the material. But we also have a very practical part. We'll be, diving pretty deep into analyzing a few microbiome samples from the human gut. And that will be done in the collaboratory notebook. So you don't need to install anything. You actually just need a browser. In this case, Google will basically front us all the computing power that we need to run through the data analysis. And you also see like some visualizations and more materials in there. And then the third part of it, and like kind of the most fun parts of that course, is like that we have a support and Q&A chat that is kind of coupled to the course. So that's like hosted on Slack. And in the Zoom chat, you already see the link to kind of join the Slack channel. So that's kind of how we will ask questions. And yeah, we will not be monitoring the Zoom chat. So please ask all your questions on the Slack chat if you have any. And the channel for the course is called Course-2022. There's also a link in the general channel when you join on Slack. So that will be basically your main resource if you have any question or doubts or just want to discuss anything about the course, or also if you have technical issues, if you can't see something on Zoom because I didn't share the screen correctly or anything else, then just let us know. And I actually am looking at a whole bunch of TAs here that are here to help, so they will be like pretty helpful in that as well. And they can tell me and yell at me if I did something wrong. So please use that resource as well. So let's get right into the weeds and run a little bit of setup, and I'll show you a little bit how that notebook is working, which is like the practical part of the course. So in the slides, you have a link to the notebook, and you also have that link if you got an email or something, so that will be helpful. So I'll just make that a little bit smaller here, and I hope you can still see my screen, but let us know if you can't. And just click the link to the notebook, and then you should see it opening up. I should be noted that you might be asked to log in into your Google account if you haven't done so. So you will need to do that to kind of use it. That will let you see and run the notebook, and you could in theory do it from here, but we actually recommend that you make a copy and that in your own Google Drive, and that will be your own copy of the course materials. You will actually save all the output and will be able to look at it later. And that can be done by clicking here on the file area. And then there is a part that says, save a copy in Drive. And if you do that, it kind of opens up a new window, which now says copy of that notebook. And you can rename it to something that is a bit nicer. For instance, I could name myself Chris. Oops. Typing live on air, always an experience. And now it's kind of saved. It's a different name. So that's our notebook, and we can actually run everything in here. So what is a notebook? We also sent you a small introduction initially in the course, but basically you can see it as like a very interactive document, which combines things like text and references, but also has those cells which actually run code, and they get connected basically to a computing backend in the background and will actually be run while you do it. So you actually do run the analysis. So it's not just, a recording or anything. The way that works is that you have that little window up there, which right now might still say disconnected. But you have those cells, they have a little play button at the moment where you start like executing one. For instance, we could do that with the first cell that actually downloads the course materials. You will see that it's connecting to the course, to the computing background. So you see that part up here, which now tells you, you know, that's your RAM, that's your disk space, and I'm running the code on some Google server in the background. And while that is running, you can also look through the other parts of the notebook. You have like a sidebar here. On top, you basically have a table of contents, but maybe more important for us is that little folder symbol down here, which actually shows you all the files. There's a folder called here materials, which is exactly where we're downloading the materials into right now. And you will see everything that we do in the course. You can download things from here. Basically, if you go to any file, you have those three little dots on the side, which will allow you to download a file. But we actually don't need to download anything because we want to run it from here. So that's good for now, but that's where we will find files later on. So right now we download all the materials and it switched to the materials folder. And we can also run the next two cells. So one that will basically just, you know, switch our working directory into the materials folder, which is all the data we're analyzing. And then we will also start running the third cell here, which actually does all the setup and installation for QIIME 2 for you. So that will just be churning along while we actually go back to our slides and look at a few more details from the course. And if you have any problems with the notebook here, feel free to ask in the course Slack channel and our TAs will help you out. So while that's installing QIIME 2, let's talk a little bit about some backgrounds on that. And one thing, like there might also, some parts of the course might go a bit quickly, or you might want to go back, or sometimes maybe things just don't run. So if you step away too long from the notebook, sometimes it resets. But don't panic, it's all good. Actually, all the output we generate can also pound in like in the treasure chest folder at the course GitHub. So even if you, for instance, for some reason you can't really participate and run the notebook right now, don't worry about it. If you want to look at any files that we generate, like the visualizations, you can always go to the GitHub later on and just download that generated file. They're all there. So there is that backup in case anything goes wrong. So that's all good. And maybe let's talk a little bit about the human gut microbiome, because that's what we want to analyze today. And I'm pretty sure most of you know a lot about it already, but just to give like a small motivation about it. So we are basically just big bags of bacteria. We are colonized by about 38 trillion bacterial cells, which are only about half a pound, or like 200 to 250 grams. So like by cell counts, if every cell in our body would vote, we would come out as bacteria, not as humans, but by weight, we have them beat. So at least we have that going for us. The microbiome is important for many reasons. A lot of the foods that we eat goes through our gut and passes by the bacteria, which are extremely well adapted to breaking down food components that we are not good at breaking down many times. But it also supplies a lot of metabolites that we are not really good at making. The microbes in our gut supply about 90% of our supply of serotonin. The microbiome has also been known sometimes to be the only road to cures like very difficult pathogen infections like C. difficile, which is misspelled here as I see. But then basically, like one of the things is just having that many bacteria in our body also helps us to keep out certain pathogens that we don't want to have living there, because those bacteria are very adapted to us. So it's actually quite hard for any other pathogen to come in and engraft in that space where we already have that complex microbial community. And also surprisingly recently, we've learned that, for instance, certain cancer therapies can be modulated by probiotics. So there is like an intimate connection also between the gut microbiome and inflammation, and that can actually like alter certain treatment outcomes. So it is a very important part, but there's many more things that could be said about the gut microbiome, and we will look at a few of them, but just to motivate that it's really interesting to study that. But how do we see bacteria? So I mean, obviously they're tiny, right? And then they're also kind of difficult to study. especially when they're coming from the human gut, because those bacteria do grow in our guts, but they almost don't grow on anything else. So like taking them out and trying to culture them in the lab is really difficult. And often if we try to do that, we enrich in a few bacteria that just tend to grow really well in the lab, and we forget about a lot of other important ones that are living in our gut, but like really hard to culture in the lab. So in the last decades or so, we have started to also bacteria using their DNA as a proxy. So it's a bit easier to just go into any complex microbial system, isolate the DNA, the total DNA from that system, and just see what you see in terms of bacteria. So kind of really assembling the DNA and looking at it. And thus, like a large part of microbiome research those days involves analyzing sequencing data, actually. So we don't really necessarily look at the actual bacteria anymore, but we're only looking at the DNA coming from it. We're trying to conclude a little bit of, what's going on in the biology from there, which has obviously disadvantages and advantages. One is we don't really see the organism anymore, but just the DNA, but also has the advantage that we can kind of see things that are really hard to observe otherwise, because we might not be able to culture them easily, or they will be de-enriched by certain laboratory techniques. So, okay, so basically what we're looking at is sequencing data, but what can we use to analyze sequencing data? Because it's not really like just a random string of DNA doesn't really tell us much per se. We kind of have to transform it into something that we can actually, analyze and see certain insights from. And what we'll be using is QIIME, pronounced like wind chime, was created around the 2010s during the Human Microbiome Project, which was kind of like one of the larger projects trying to map out the microbes in the human gut. And that was under the leadership of Greg Caparaso and Rob Knight, who is probably really well known for being one of the founders of the microbiome field. And was basically, observing in the Human Microbiome Project that, there's certain analysis steps that are pretty specific, and it was also kind of challenging because you have all those DNAs coming from a lot of different organisms at once in a single sample. There was a lot of practices and good practices you kind of had to go through to get like reasonable data out of that. So they kind of tried to couple that up into that kind of software package collection, QIIME, that should make it accessible to a lot of researchers all over the world. So what is QIIME? Well, it stands for Quantitative Insights into Microbial Ecology. And officially, it's a powerful, extensible, decentralized microbiome analysis package with a focus on data processing and analysis transparency. In more practical terms, it's essentially a set of comments that is like bundled, that you can use to transfer microbiome data, like sequencing data, into intermediate outputs and visualizations. Its most common use is via the command line, but there's also other interfaces to it. Though there's like a web-based interface, and it's also integrated into the Galaxy framework, which is sometimes used for analysis, which kind of gives you like a more decentralized computing infrastructure. But in most basic way, it's a set of comments you can run on your data and generate like intermediate data, and then finally also generate visualizations. So QIIME 2 was introduced in 2016 and improves upon QIIME 1. So basically, QIIME 1 was used a lot by a lot of people. Basically was one of the first kind of really maintained software packages, large ones to kind of analyze microbiome data that was kind of accessible also. But there was a lot of like user experiences that kind of went into designing QIIME 2. Like one thing was kind of making sure, so one thing with QIIME 1 was that it would just generate the output files, and it was usually really hard to tell where those files came from or parameters were used to generate them. And then there was also that question of like, is it feasible? Can I plug in certain data types into like a certain type of analysis? For instance, if I normalize my data a certain way, is it still feasible to do that statistical analysis on that? So they actually have something which is called the semantic type system, which can actually check if your input is valid for the particular type of analysis, kind of really ensuring that what you're doing is kind of, best practices and compatible with what the field is saying. There's an extendable plugin system, so it's quite feasible to write a plugin for QIIME 2 if you have any new methods that you want other people to be able to use. And there's multiple user interfaces, like I mentioned, which some of them are still a little bit in progress. I think one of the strongest aspects of QIIME 2 is actually the community around it. It has like an awesome community. It has a wide range of tutorials. There's a lot of courses like this one here, but also others. Has a really good documentation, and like one of the superpowers, I think, of QIIME2 also has a really good user forum that is super accessible, friendly. There's a lot of people that are kind of looking through that and can help you. So I would definitely recommend you to use those resources if you ever like get into the weeds of the QIIME2 analysis for your own data. And let's talk a little bit about like how QIIME2 works, what is like the abstract design of it. So QIIME2 manages something which I call artifacts. Artifacts are basically intermediate data that you generate. So usually kind of we analyze data in something called more or less like a pipeline. So you start, for instance, from your raw data, then you transform that into something else, and you do another method on top of that that really transforms it into something else. And then at some point you end up with, you know, visualization, a plot, something you can put in a paper, for instance. So it basically manages actions and artifacts. Artifacts can be transformed into other artifacts via actions. And then at the end, you have something called a visualization. Visualization is kind of an endpoint meant for human consumption. It doesn't actually have to be like an image or figure. It can be, for instance, just you know, a table that like summarizes statistical results. But the idea is like a visualization is something a human has to look at to interpret it. Whereas artifacts are usually things that a human can't make much sense of, but which are necessary to kind of generate. So like that's kind of shown in a very abstract sense here on the left side of the slide. So you have those artifacts, and then you have those pipelines. Pipelines are just chains of transforming artifacts into each other via actions. And then you have those visualizations that are generated by visualizers, which are the final output. And like on the right, you have a specific example how that might look like. So you might have raw sequences. You might have to split them apart into the sequences for individual samples first because they were pooled on the sequencer. Then you kind of want to denoise them, like kind of remove like sequencing errors and to recover original sequences. And then you might want to assign taxonomy and so on. All of that generates in the individual intermediate artifacts. And at the end, you might have some differential abundance testing or statistics or plotting, which are the visualizations. So the main idea is that you can build up those pipelines pretty easily with QIIME2, and we will see how to do that in a second. So like I said, the only thing to remember here, or one important thing to remember here, maybe not the only thing, is that artifacts represent intermediate steps. So that's something that's quite hard to make sense of usually, but visualizations are endpoints. So they're for our consumption, we can look at them. And maybe, you know, because that looks really abstract, and we will generate some weird sounding files that end in like QZA or QZV, but those are all just zip files. The main idea of QIIME2 is also that the data it generates can actually still be understood and used later on, even if QIIME2 would suddenly cease to be, which is not going to happen. But like those things are not like something that only QIIME2 can work with. Basically, any QIIME2 artifact is honestly just a zip file, which has a lot of annotations, but it has a data folder that just contains the actual data file, which can be a CSV table and so on. So you can always generate that original data again if you wanted to. Okay, so what we'll be doing with QIIME2 is Amplicon sequencing analysis. So let's maybe talk about that a little bit. What is the difference between Amplicon sequencing and, for instance, shotgun metabonomics or shotgun sequencing? Well, the main trick of Amplicon sequencing is that I don't actually sequence a full genome. I don't even sequence a full gene. I actually only sequence a really small part of a gene that's very informative to us. Those small parts of genes usually get amplified via PCR, and that's why it's called an amplicon. It's a small amplified region from a gene. The basic steps that you go is that you extract the DNA from the sample. So usually you have a method to physically kind of rupture the cells. And then you have like an entire protocol that pulls out the DNA from those samples. You amplify those small regions that you're interested in, and then you usually sequence them on a sequencer. And those regions are usually designed in a way that every single read on a sequencer, or at least the pair of reads on a sequencer, covers the full region that you're interested in. So you kind of have that really high efficiency sequencing where like every single read kind of covers the full area of interest. That means you don't really need that many reads in sequencing. So you kind of can go and put a lot of samples on a single sequencing run. So it's very cost efficient, which is why it was, until fairly recently, still a very, and probably still is the most prevalent microbiome sequencing technique that you will see. The limitation is, so it is not genomics, right? You actually get only a small amplicon. Usually the only thing you can really do with it is kind of look at the variability in that small amplicon, maybe infer taxonomy, like we can try to assign names and see how many different bacteria were there, which is already complicated with that data. But we don't really get any information about which genes were in that genome. So all of that information isn't there. So if you wanted to do that, there's shotgun metagenomics. It's actually a bit simpler in the main idea. The main idea is that we take all the DNA in the sample, we chop it up into really small parts, and then from that we try to reassemble it into the full genomes of all the bacteria that were in the sample. Obviously, since the genome was much larger than the amplicon could be something like 200 or 400 base pairs long. A full genome of a bacteria is 2 million base pairs. We need much more sequencing depths, so you cannot pool as many samples in a sequencing run. And metagenomics is intrinsically expensive because of that. And also needs a bit more complicated protocols, because if you want to, sequence random fragments on the sequencer, you kind of have to need methods that can actually add adapters to those easily, and those are random sequences, so you kind of need adapter kits and so on. So it's a bit more, it's a bit more work, and it definitely also needs a bit more deeper sequencing to get anything sensible out. But the good thing is, if you have an abundant taxon, you might pull out the full genome, giving you a lot of information about the genetic content of that particular bacteria. So if we talk about amplicon sequencing, we often use the 16S gene. Why do we use the 16S gene? Well, because it's almost like somebody designed it to be used for amplicon sequencing analysis. So the 16S gene is part of the 16S ribosomal subunit. It's essentially the part of the ribosome that recruits the mRNA to the ribosome. So most living organisms actually have the 16S subunit on the 16S gene. Especially all bacteria have it. So that's really nice. We have that very conserved thing that we will always be able to find in any bacteria. But then it also has those interspersed regions of very conserved regions, which will always have almost exactly the same sequence for all bacteria that we know of, interspersed with very hyper-variable regions that differ quite a lot between different species, genera, and so on. So they're very diverged. So you can basically identify the name of a bacterium based on those variable regions. And there's, depending on what you're studying, certain regions are better. The most common ones for bacteria, some were the V3 to V5 region. V3, V5 is pretty common. V4 is also very, very common. It's a bit shorter. Depending on how long your reads are on the sequencer, you usually design it based on that. But yeah, that kind of makes it perfect, because if you want to amplify anything with the polymerase chain reaction, you need that initial kind of primer pair that fits really well. So we can basically target the primers into the conserved regions, knowing that those sequences will not be fairly different across bacteria. And then they get elongated across the hypervariable region, which actually has the information that we want. So it's kind of really designed for, putting in primers around them and then amplifying those variable regions. They're also fairly short, so they're kind of perfect for short read sequences that we do it. So that's what we'll be doing, basically. And let's talk a little bit about also the data sets that we'll be looking at. So the advent of cheap sequencing has generated a lot of data. But is that data really representative of the, the microbiome on this planet, or the gut microbiome that humans have on this planet? And the answer is, well, not really. It's actually just, and Sean alluded to that already, a few countries that account for the majority of microbiome data. So it's really not representative of the populations that we have. Most of the countries on this planet are actually not well represented. And there's a lot of reasons for that, and a lot of biases. So one of the main reasons is basically the data get generated where we have basically the financial resources, institutions, or research teams that actually studies that, which are very enriched in Northern America and Europe. There's also institutionalized biases that kind of can skew that towards that overrepresentation. And this information actually propagates to a lot of different data sources. So it's not only that we see that most of the samples that are generated come from, those overrepresented groups, but then It's also that this gets propagated into reference databases, functional annotations, and so on. If you look at reference genomes, you will see that, especially from the human gut, you will see that they heavily cover those bacteria that appear in those populations that are already enriched. And you will not find a lot of really good reference genomes for other populations. And you know, there is much more to say about that, and you will hear more about that also in the symposium talks, which are like all recognized experts in this area and can will tell you much more and give more background on that as well. But it is a little bit of an issue because most of the data isn't really coming from all over the world. So who's studying who? We'll be looking at a total of 9 samples today from 3 communities, the Hadza in Tanzania, the Chepang in Nepal, and the Mefa in Mexico. All of those are indigenous populations, which, you know, sometimes had to adapt their lifestyle simply because they have been encroached by like modern settlements, not because they wanted to. They all have very different lifestyles, so there's a very strong there's different cultures and also lifestyles here that act in those 3 different populations. But one thing we also have to kind of account for when we talk about that is not only, you know, who is being studied, but also who is studying who. So all of those manuscripts studied indigenous or marginalized communities. None of them were necessarily led by members of the community per se. So often those studies are conducted by foreign institutions. So even though indigenous communities are studied, it does not necessarily mean that the interests of the community are represented. And as a really good like kind of guideline, Emily Whistle contributed that like awesome like infographic here of questions you can basically ask yourself when you look at microbiome studies to kind of see, you know, how was the interaction with the community actually happening. So for instance, you can ask, was the community really early on, was the community involved in the study design and collection? Was the question asked actually relevant to the community being studied? Then an important aspect is also who has the ownership of the samples and data later on. You know, was the community reimbursed? Like a lot of those studies might actually generate quite a bit of risk for the communities. The question is, was there like any way to make good of that, or whether rewarded for their involvement in the study? You can ask if the research team tried to train anyone from the community to actually analyze the data that was generated. So whether participated in generating the research output, and then is that reflected in the final manuscript that is put out, right? Did the community participate in writing up the results and interpreting them? And is anyone from the community also on the author team? Well, those are like really good questions to ask if we look at the study. So we should be aware that there is that social context of studying marginalized communities as well, which we have to take into account. And again, there will be more about that also in the symposium, but I also want to make a small shout out to the micros and social equity working groups that was recently founded, and you can click on the link and get some more information. And I think there will also be like a speaker series in the beginning of next year, so keep your eyes peeled for that as well. And there's also some references to get some more background on that as well. So what we'll be doing today in a very broad sense. So basically, we'll be looking at the sequencing data, the raw sequencing data. The first thing we will do is we actually will trim and filter it. So we kind of look through the quality and which of the raw sequencing data is actually amenable to be analyzed further. Then we do something which is called inferring sequence variants of the Amplicon. So basically, a sequencer is not a perfect machine. It will generate a lot of reads. A lot of those reads will have errors. So there's always the question like, how many actually unique organism did you have in your sample? Because you might see millions of reads, but there might actually just be a handful of organisms in the sample. And just because of sequencing errors, the available sequence space looks way more diverse than it is in that sense. We will look a little bit on how we can assign taxonomy to particular sequence variants if we have identified some. We talk about normalizing data so we can use it for analysis. And we will also talk about ecological diversity metrics, kind of more global descriptors of the system. And all of that is aimed at generating ecological insight from that sequencing data. And before we really jump in, let's really talk about what is like the first data that we get. So Usually what we get, here's an example from Lumina. So, but usually the data that we get from a sequencer are saved in something called FastQ files. So this kind of means faster plus quality. Faster is like an old format to store just DNA sequences, which is very simple. It's just a text file, which essentially has a title and then the DNA sequence. And the FastQ files add like the quality information to that. If you, for instance, use Lumina sequencing, often you get those really longish sample names that are shown here, which you can kind of decompose into that. Usually you have like a sample ID. So that's something usually that you choose, or your sequencing unit will assign if you use a sequencing like shared unit in your institution. You usually get like another ID system, which is like the sample order. That's kind of is helpful because it tells you in which order the samples get injected onto the sequences. So sometimes it can be helpful to see if there's certain biases or errors there. You get a lane, which, you know, until recently, you would usually just see a single lane because those the flow cells in the sequencer are often used only one lane, or you would like put on one pool on a single lane. Those days, most of the sequencers parallelize that. So like, and those sequencing like flow cells basically that are put on the sequencer and that connect to the DNA and sequence it basically are separated into multiple lanes those days. So you might see other indexes that tell you which lane was kind of used for that sample. And then you also see the redirection at the end, which is usually, you know, it could be only a forward read if you do single end sequencing, but if you do paired end sequencing, you will get the indication of which direction was read from as well. And then you usually, it ends with a 001, which is always the same. And then often they're compressed as well. But that's kind of the file name. What does it look like inside? Well, you get like the title, like a name for the sequence denoted with an @ Usually that's just your sample ID and then like the index of the read. So in that sample would be that particular sample ID and would be the read number 13,917. It might also indicate the length of the read, but those things are optional. Then that's followed by the actual DNA sequence of that read that was sequenced. Then there's a plus as a separator, and then you get a really random string of like different letters and symbols, which is essentially the quality encoding. And why does the quality encoding look so weird? Well, it looks so weird because if those were actual numbers, like for instance, a 10, then it would be hard to map that to the individual bases because each single base in this DNA string has a quality score. But if you would write a 1, 0, you wouldn't actually know, is it the T with a quality of 10 or has the T a quality of 1 and the A has a quality of 0? So it would be misaligned basically with the sequence. What we did is we kind of just selected like random symbols, like a lot of them, and then we kind of order them up and use that as a sequencing quality assessment. It's really sad that this was designed before the emoji revolution. Otherwise, we might have gotten an emoji-based quality score. So, you know, it would be much more fun to see like that the first base has like a vomit emoji, and then the next one is like a heart emoji or something to judge qualities, but kind of missed that chance, unfortunately. So we have like random letters now. But basically, you can pass those letters back in and assign a quality score to those bases. Which brings us to, you know, a kind of a practical question if we want to analyze data with QIIME2 is, okay, we have our raw sequencing data, but then QIIME2, the only thing that QIIME2 does is transforming artifacts into other artifacts, right? So like, how do we get from the raw sequencing data to the artifact? Looks a bit like a chicken and egg problem, right? So we can only get artifacts into QIIME, but we have raw data, which needs to be an artifact. So how do we do that? Well, that's what we will be looking at now. So let's actually switch back to our notebook and actually get cooking. So you should still have that open. And you should have seen that the installation of QIIME2 has finished, and it should tell you that everything is okay. If it isn't, please ask in Slack to get some help. Or if you haven't run it, then that would be a good time point. But yeah, it should have taken about 10 minutes, so more than enough because we talked much longer. And that's an overview again, what we'll be doing today. But the first thing we'll do is actually resolve that chicken and egg problem. So we need to get our raw data into a QIIME2 manifest, QIIME2 artifact, sorry. And we'll use a tool called, which is basically a manifest import, which is like a tool we can use to kind of exactly resolve that problem. So it takes raw data with annotations and generates the first artifact that we can push into a QIIME2 pipeline. And just to give you an idea how that looks, if you look into the data folder here, you actually do see the manifest. So if you would look at that, it kind of looks like that here on the side. And it basically just maps the ID to the particular files. And the WLPVD looks really cryptic, but that just means that's the current working directory. And it tells you that's in the data folder. And for instance, for the first sample, that's the forward reads, that's reverse reads. And that kind of tells QIIME how to connect those samples to sample IDs. It's actually quite practical to use that format because that manifest is pretty similar to things you often have to attach to publications. Or if you want to upload your data to a public repository, you have to basically provide something very similar. So often you have that already available. So how does like the QIIME2 comment look like? In that notebook, like every QIIME comment will be prefixed with an exclamation mark. Just means we want to actually run a comment in like the comment line. By default, it would assume that everything is Python code in here because it's a Jupyter notebook and it's made to run Python as a programming language, but the exclamation marks will run it as a comment line comment. If you run it on your own computer, just in the terminal, you can just strip that exclamation mark away. So you would start with just QIIME. And then usually what we will be doing is basically use an import tool, which, a lot of that comment looks very cryptic and we'll be like, unraveling that in a few seconds. But basically we're telling it we want, we have paired end sequencing with quality scores. That's kind of the type. Like I said, QIIME is a type system. So the initial artifact needs a specific type assigned to it. It needs the manifest of the data and it will generate a QIIME2 artifact. And usually those files end in QZA. And then we also have to say what the input data is. And like the only thing we say that's paired end data, it has a manifest and that cryptic thread 33v2, that basically is a kind of encoding for the quality scores that was used. That would probably work for most data sets those days. So if we click on there, it will basically start importing the data and generating the first artifact. And you will see that pop up here in the materials folder. If you don't see it pop up after the comment has finished, sometimes you need to kind of push that refresh button here and then you will see it. So then at this point, we have that first artifact that we can actually start plugging into QIIME and to kind of, decompose that a little bit. How do QIIME2 comments actually look, right? They always start with QIIME and then you specify a plugin, which is like the collection of actions or tools that are can be used for usually a fairly common purpose. You specify the name of the action, and then you have a lot of arguments that you pass in with --. Then there's often a single letter and then the name of the argument. And that first single letter is usually kind of what the argument means. So if it's an I, it means it's always an input. So that's something you already need to have. If it's an O, it's the output. So that's the name of the files that you want to generate with that action. If it has a P, it's a parameter. So for instance, sometimes if we do quality control or analysis later on, we want to specify certain parameters, say what's our quality threshold and so on. That gets passed in with P. And if it has an M, it's metadata. So annotations, for instance, for the samples. So if we go back to the first tool, that's what we see, right? We had QIIME, the plugin that we used was tools, the action that we used was the import action. It had a type argument, which is usually not common, but that's only for the import part. And then it has like input, output, and so on. And then we can also kind of look at another comment just to kind of drive that home. And that's like an action called summarize that comes from the dmux plugin. And then it takes as input the sequences that we just generated, and it does create our first visualization called qualities. And that will actually tell us the sequencing qualities. So we can actually pass in all the quality scores and look at what, how does our data set actually look like. So let's run this as well. And then let's look at the actual visualization. Might take a few seconds. And yep, just again, if you have any questions in the Slack channel, you will get help. So that will actually generate a file that ends in QZV. That looks pretty cryptic. So how do we actually look at it? Because it's meant for us, right? So we should be able to look at it and draw conclusions from it. And to do that, there's several ways to do it, but like maybe the easiest is there's actually a website on the QIIME website that is a viewer. So we can open that in a new tab here that looks like this. It basically just asks you for a visualization. So you could click on that and upload it from your computer. Right now, it's still stored here in our notebook environment. So what we can do is we can maybe refresh to see if it pops up. So we have that qualities QZV file here. You can click on the 3 little dots next to it, go on download. that will download that file. And then usually you can just drag it from your downloads. Oops, like that. So if the download is still open, well, actually, this is not working in my case, but let's do that again. While it's still showing up down here, you can usually drag it over here on the visualization directly, and it should open it up. If that doesn't work, just to go back, you can also click also on that large area here, then go into the downloads folder and then just look for the file and open it up. And yeah, that might look slightly different depending which operating system and browser you use, but like that second strategy always works. The first one also should work, but you might, you still need that view of the download open. So what does it look like? Well, you get a summary of the sequencing data first. So it tells you kind of across the samples, what's the minimum or maximum number of reads that we had per sample. So reads are those like initial small DNA reads that you get from the sequencer. Usually for 16S Amplicon data, we kind of say that everything above 10,000 is good. So we can usually characterize the sample pretty well with that. The more the better for sure, but 10,000 is perfectly fine. In that case, our minimum read number across all the samples is 11,000. So we're definitely having that. Our mean is actually like around twice that. And we have some samples that have up to, almost 200,000 reads here. But yeah, so we have like a lot of reads in the samples, so it works pretty well. You also get some summaries across the samples, what the reads are. Sorry, no, the total is 192,000. The maximum is 34,000 reads for a sample, which is perfectly fine. So you can look at that, and we kind of see the quality overview here. Maybe more interesting, though, is to click on that little tab here, which has interactive quality plot, which actually tells you the quality across the read. So in this case, the total read length was somewhere between 150 base pairs and 250 base pairs for most samples. So you see it going up to 250 base pairs. And what that basically shows you is for every read position in the forward and reverse reads, you kind of see the quality score. So what does it actually mean? We have quality scores here from 0 to 40. Higher is better, but it is actually in logarithmic scale. So there's kind of rule of thumb, like initially, like put out by Illumina. So if the quality is 10, that means that you have an error rate of around 10%, which is pretty high. So that means 10% of all the bases that you see in the read would be kind of random errors in sequencing, which would be a pretty high fraction. But then if you go from 10 to 20, 20 actually only means a 1% error. So you kind of get like an order of magnitude less errors or more accuracy. If you go from 20 to 30, you have 0.1% errors, and 40 is 0.01% errors. So that's really, really small. Actually, with 0.01% errors, you would actually expect that, most of the reads are actually perfectly error-free, which is not really true, but that's kind of like a guideline. So usually, like if you see something above 30, that's really good. Above 20 is still kind of usable, but you don't want to dip it kind of in the 10 region. And we do see that, we see kind of, that's a pretty typical distribution. We see the qualities decline towards the end of the read. That's kind of like a consequence of the physical limitations on the sequences. So it gets a little bit less accurate at the end. You also see a dip in the beginning. It's not necessarily like really lower quality, it's just that those quality scores kind of take also the previous bases into account, and that doesn't really happen at the beginning of the read, so that influences it a little bit. And you often also see much worse qualities for the reverse read, so that's pretty common as well. So we kind of maybe want to trim that a little bit, cut off a little bit from the reads, from the 3' ends of that read to kind of keep the quality high. So usually here we could be fairly permissive about the forward read. Maybe 250 would be a good cutoff, and then we would be a bit more strict because we do see that, the last few bases in the reverse reads are basically complete nonsense. So they're kind of like random sequences. They have nothing to do with what's actually sequenced. So we would be more aggressive here, maybe cutting off at 200 or even earlier. But one thing also important to check out is this. You get like a length summary at the end that tells you, how long were your reads actually. And you do see that even though we have many samples where the read length is indeed 250 nucleotides. There's also a bunch of samples in there which only reads of 150 nucleotides. You will usually not see that in your own sequencing runs. It's more like in that course data, since we kind of combine different data sets. But that kind of limits it. So because there is like one surprising thing later on, if we tell QIIME2 that we want to trim a read at 250 base pairs, you might expect that if my read is only 150 base pairs long, nothing will happen to it and gets passed along in the analysis. But that's not what happens. It actually gets thrown out, because trimming at 240 means the read has to be at least 240 base pairs long. So we will actually be trimming a bit more aggressively at 150 nucleotides for the reverse and the forward, which kind of cuts it off into really high quality areas. So it will still be fine, but that's like something specific to the course data. Usually you use the quality profile to guide your cutoffs. You try to find a cutoff where you approximately above 20 maybe. But that was, already pretty informative. First look at our data. And maybe one thing to show you as well, that you have those extra annotations. So you actually get the citations for the methods that were used. And you get that provenance, like I said. So every single thing generated by QIIME has a full history of how it was generated. So you see here, that's the visualizations that we did. And then before that, you actually see also our import that told us which files were used, which files were imported, and so on, and what was the machine that ran it and things like that. For later files, those things will look pretty large. So you have the full pipeline basically like still encoded here. So from any single file in QIIME, you can always reconstruct the entire history that was taken, which is nice, because even if you have nothing else later on than this file, you will be able to see what's going on. Okay, I'll be going back to the main page of the viewer here. If you closed it, you can also reopen it with a link later on. You can use that to look at other visualizations. Okay, so that was the first look at the data. We got our data imported. We looked at the qualities and kind of know where we want to cut off the reads for the quality filtering. And we will jump to one of the larger parts of the analysis, which is called denoising. And we will talk about that in a second. The only thing we will do now is we'll start doing that process because it takes a little bit to run. It's a somewhat complex algorithm. But just to understand like what we will have to put in, so it's that is like a plugin called Data2, and we will talk about that in a second. It has an action called denoised paired. So we want to denoise the data, and we have paired end data, so that's the action we want to take. Our input is again the sequencings that we imported initially, so our sequencing data. And now we already decided that we want to truncate the forward lengths at 150 base pairs and also the reverse length. So that's what we'll be doing here. That's basically so we can use all the samples that we had. And we will also use two CPUs here to just make it a little bit faster. We specify an output directory. So the Data2 denoise paired action actually doesn't output only a single artifact, it outputs a bunch of artifacts. So we will save them in a directory called Data2, and we use the verbose options, so it actually tells us what's doing. So we can run this. This will start running here, and it will take like a little bit, which gives us ample time to jump back into the presentation and go ahead with the explanations here. Okay, perfect. So we ran our first comments and we brought out the big guns now with Data2, which actually does quite a lot, like a large chunk of our analysis. So it will filter and trim the reads. I can kind of also throw out like sequencing reads that have like globally very low quality. And it will find the most likely original sequence in the samples, ASVs, and we'll be looking at what that means in a second. It will remove chimeras. Again, we'll be looking at that in a second. And it will actually count the abundances of each of those variants. So we'll basically kind of try to decompose that into individual 16S Amplicon sequences that were in the original samples and tell us how much of each of those sequences were actually present in the sample. So in the pre-processing, we kind of looked at it. It actually does a bit more things that we talked about. So it does trim the low quality regions. It removes also reads with low average quality. It also removes reads with ambiguous bases. Sometimes the sequencer might not even be able to call a base, and it might just put an N instead of like a CGT. And it also removes Phi-X sequence. It's a bacteriophage small genome that has to be added to Amplicon sequencing runs because modern sequences are not really good if your sample is really low complexity. It has something to do with the fact that they're kind of reading that via like a fluorescent signal in the end, and that gets saturated if you have too many small fragments that are just too similar. So usually you add in just like random DNA from that particular Phi-X genome here to kind of increase the heterogeneity of the sample and makes that work, and it will do all of that for us, so we don't have to worry about that. And then what are those ASVs? Well, those ASVs are called Amplicon sequence variants. And it's actually what we, supposedly had in our original sample, right? So when we amplified those regions with PCRs, we had, for each specific 16S gene, we get like a single sequence out of it. But like I said, what happens in the sequencer is that it sometimes makes errors. So basically, like a single sequence in the original sample might generate a lot of reads that look slightly different. And that happens to like every single unique DNA sequence that we had in the sample. So it's a bit of a chicken and egg problem yet again, because, if I kind of would know like how often that happens, I might be able to kind of deconvolve it into the original sequences, but I don't. If I knew which was the original sequences in the sample, it would be fairly easy to map which are, just read versions of it with errors, because I would see that they're really similar, but they have maybe just a single base error. So that kind of chicken egg problem in bioinformatics often gets resolved with something called the expectation maximization algorithm that kind of especially deals with that kind of problem. So you kind of have like groups, but you actually don't know which of your data points belong to the group. And you have like an error model, which you also don't know. So basically it kind of does like a back and forth thing where it first kind of assigns things to random groups, then fits an error model, then uses that error model to assign them into new groups more consistent with that model, then recalculates the error model, and at some point it converges and you get like the real signal out of it. And what does an error model actually mean here? Well, the error profile means that, based on the quality score, we might actually have very different probabilities of seeing a certain transition. So for high quality, we might see, that's a probability of certain transitions is kind of not the same. So for instance, like reading an A as a T might be less likely than reading an A as a G, and so on. And it basically generates those profiles based on the quality score or the inverse quality score. And then at the same time, after it has fit those models, it can actually identify what is the original sequence that generated all those reads. So it's like a really cool program that's, we didn't always have it in the microbiome field, but now we have it, and it's like pretty powerful because in theory, it gives us back the original Amplicon sequences, and you're getting a little bit of like Seattle street noise here as a bonus as well, which is nice. So we can kind of assume like that more or less what we get out is like, even if you see in your sample, you might have 10,000 unique reads, this might actually just group into maybe like 15 Amplicon sequencing variants, which in theory represents the exact number of 16S variable region sequences that we had in the sample. So that's like really powerful to analyze. And then one thing we should also, like again, and that's kind of like owed to the fact that we're kind of amplifying extremely similar regions. So like I said, there's a very conserved region, the 16S gene that we attach a primer to, but that also means that, most of the sequences that we kind of want to amplify here are very similar. So mispriming is really common in polymerase chain reactions. So essentially, when it kind of does that extension where the primer attaches and it starts kind of elongating the read, it sometimes aborts the extension, can be because, the cycle ended or the enzyme fell off and so on. The problem is that those like partial reads often, are a big part of the conserved region of the 16S amplicon. So it's really easy to have mispriming. So a completely different 16S sequence goes to it, then gets elongated again, and you get something called a chimera. So it's basically a single region that is actually a hybrid between two variable regions. And that can be like an issue we want to kind of resolve for that. And DALA2 actually has a filter for that, where it can detect which of the ASV are just chimeric versions and removes those as well. So one question we can also ask ourselves here, and we will look at in a second, is the primers used here were F515, R806, and those are the exact like positions in the 16S gene. So how long is the amplified fragment really? And, do our reads still cover it completely, right? That's like a question we might ask. So we get from that like a table containing the counts for each ASV in each sample. We also have a list of the exact sequences already recovered. So what could we do with those two data sets, right? What quantities might we be interested in? Yeah, and feel free to post some ideas in the Slack channels. There's definitely some, very low-hanging fruit, but also like some other question we can ask about it. And one thing that we could look at, even if we don't yet have like taxonomic annotations, is just, well, how do the organisms in our samples relate to one another, right? Because we now kind of have that idea that every single amplicon that we read is kind of related to a single organism. So if we get 15 amplicons, I mean, sometimes an organism can have several 16S genes, which makes it a bit more complicated, but like more or less like rule of thumb, they're kind of relating to a single organism. So if we compare them, we can actually also see how many of the organisms in our sample of the microbes are actually very similar to one another, or some very, phylogenetically distant ones. And to do that, we can build a tree. So we can now kind of take all the ASVs that we get from our samples and align them into a phylogenetic tree, which are usually built from multiple sequence alignments. And there's a lot more that can be said about that, but just as a basic idea, how would we map out, for instance, evolutionary distance between sequences? The main idea is, well, I'll talk a little bit slower, is that we take multiple sequence alignments, we align all the sequences to each other, and then we will sometimes see that, two of the sequence might only differ in a single nucleotide, or there might be, differ in a lot of the nucleotides, which is kind of related to, evolutionary distance, like when have they diverged. So we can use that as a similarity measure on a tree. And we can visualize that as well. We will look at that in a second. So let's maybe go back to the notebook and have a look. So our data to pipeline ran, took about 7 minutes in my case, should be pretty similar for you. It basically did exactly what it said. It learned the error rates, and then it denoised the sample, removed chimeras, and so on, and wrote the output. So now we have that data2 folder with a whole bunch of artifacts in here, one of which actually contains the actual sequences of the amplicons that we recovered. Then a table that also quantifies how abundant each it is, and some denoising stats as well. If that didn't run, then we could also have used that comment here. That will do nothing if you would run it now, so you don't need to run it if your comment worked, but that would basically just take the burden from the treasure chest. And one question I asked is basically, do we actually cover the full amplicon? The question was like, if our, full amplicon has a length of around 290 base pairs, which is, implied by the 550 forward and 806 reverse position. Because one thing that data2 also does is merging the amplicons after it has denoised them. It also merges them into the full amplicon. But that only works if the reads actually cover the full amplicon. So usually in sequencing, it's always designed that you have like an overlap. So it should, in theory, cover the full region, and you have that overlap so you can basically join those reads together at the end. But the thing is, we do quality trimming, right, which takes off some of the 3' end. And if you do that too aggressively, sometimes you get those cases here where there's no overlap anymore, and you're not able to merge in back to the full amplicon. And you will actually get really no output from data2, like it will tell you that nothing is there. So we usually want to make sure that we still have a little bit of overlap. It doesn't have to be as long as in the original sequencing, but it has to be maintained. Luckily, the B4 region isn't that long. With 250 base per reads, you would basically cover it almost with any single read. But the pair would have massive overlap of more than 100 bases. But since we cut them down to 150 base pairs, we still have enough overlap, like around 10 nucleotides, and that's enough to merge them back together. But you can also adjust that overlap parameter, how much overlap you need at minimum to merge them back together with the data2 pipeline in case you have very little or a lot of it. And we talked about the chimeric data. One thing we can do is just double checking that, what happened to the reads as they pass through the data2 pipeline. There might be cases where we actually see a lot of them being lost as being chimeric, for instance. Maybe they couldn't be merged because, the quality on the 3' end was so bad that it couldn't actually identify, like merge it back together into the amplicon and so on. So a good check is, do we still maintain most of our data after all those quality checks and transformations and so on? And we can use like an action called tabulate that takes those statistics that we get from data2 and generates the visualization, which in this case is not an image, but just a table. So we can go through that and generate that new visualization also in the data2 folder. So it will kind of appear in here as a force file in a second. And we will have a short look at that And you can also investigate a little bit further if you want. So same strategy, I download it. I go to the QIIME2 viewer, and then I pull it over, drag and drop it over here. And what it will tell you is exactly for each of the samples that we had, it will tell us how many sequences do we have as input, how many reads did we have. Then after quality filtering, what was left? What's the percentage? And we see that, you know, the quality was really good in those sequencing. More than 90% of the reads were actually maintained. Then you see after denoising how many of the reads were kept, because there can be certain cases where Data2 just says, oh, I can't really assign an Amplicon here. So I throw out those reads. They just look like they're not really making sense. But again, that keeps the majority of reads here. And then after merging, that might drop a little bit. Like I said, if that overhang isn't enough or it's really low quality, they might not be able to be merged. And we do see some drop here, but still more than 80% are maintained, and that's fairly normal. There's always some reads which have like some errors in the 3' end, or you read into an adapter or something like that, and they can't be merged well. There's a lot of things that can go wrong when like those small fragments get sequenced, so you do lose a little bit here. And then you see like what is the final read number after removing all those chimeric sequences, which drops it maybe another 5 to 10% here. It might go as high as 10, 20, 30%. It's really dependent on how you run the amplification. So that's really specific to the experiment, but it's not unusual to lose like a certain fraction here. But those were all like sequences that were chimeric. They're not super informative to you anyways. But in the end, we keep around 75%, 80% of the data, and that's kind of the important thing we care about here. So it's still pretty good. So we can still get a lot of information out of that. Or maybe leave that open. Just go back to the initial page here. Okay, so we know that everything worked out well. We have our Amplicon sequences. And we have them counted, so we know how much of each of them, because some of them will be very abundant. If the organism that contributed that 16S sequence basically is very abundant in the sample, you kind of also see more of the reads mapping to it. So it kind of helps us to kind of also see how much of each of us there. And then, as we said, like one thing we can look at is how similar those are. And we do that by basically doing those first two steps. We generate an alignment. So those sequences are really short, so we can align them pretty quickly, even if it's a lot of them. So we will do that on the generated Amplicon, so the actual regions in the 16S genes that we identified. So that's the first step here that we can run that kind of generates that alignment. Again, it would be something that's really hard to look at. It would be like a massive multiple sequence alignment of every ASV that we have. And then we can kind of use that to generate basically a tree representation of it, because those alignments kind of tell us how similar any pair of Amplicons here in the sample was. So we kind of can use those trees. There's, again, a lot of output. So one is the alignment, then there's a mask alignment, then there's a tree, and there's also a rooted tree. So the difference between a tree and a rooted tree is that a rooted tree is assigned basically a center, like a single common ancestor for every sequence, whereas a tree doesn't really has to have that. The rooted tree is just a little bit easier to visualize sometimes. And we will use like a tool called a plugin called Empress, which has a lot of really cool visualization techniques for phylogenetic trees. And we will just, for now, we will only show the tree and look at it. And it will not contain as much information, but it gives us like a structure of the Amplicon sequences in our sample. So that might be interesting to look at for us. So it generated the tree. We have that tree folder here. So you should basically see that Empress visualization here. It's the visualizations always the files that end in a QZV. And we do the same thing as before. We download it, we drag and drop it to the visualization. And bam, we have a phylogenetic tree. We can zoom in. You can click on the end nodes, and it will actually tell you something very cryptic. Why does it have such a cryptic name? Well, basically, those Amplicon sequences are pretty long. So one thing that QIIME does is that it just assigns a unique ID to each of them. So basically, you just see, oh, that's the Amplicon that got that particular ID, and it tells you the node length. there's a lot of options here, and you will actually go through that again with a lot more annotations later on in the exercise to actually annotate the tree and really look what taxa you have in there. But just to give some examples, you could basically change the layout. You could go to the circular layouts that we see in a lot of publications. You could start making it ignoring the branch legs, or even making it ultrametric, which gives you those kind of circular layouts that you see often. And again, you would still be able to identify the individual nodes. And you already see there is quite some patterns here, right? So you have things like there's basically one really early divergence to some of the ASV versions here, if we draw that to the line. So those seem to be fairly different, and you can look later on what that actually means. But you also have those groupings here, right? Those kind of clades that separate pretty late. So that means it's a large group of fairly similar sequences, which a particular substructure is still in them. So that's something we'll be interested in looking at later. But that's like gives them an information, and each kind of outer node on that plot is a single amplicon sequence variant. And even though there are a lot here, you can also see they're definitely not the total number of reads in our sample, which was around 300,000, right? So we do see that we kind of reduce that with the denoising step. I'll be going back to the main page. Again, if you close that tab by accident, you can always reopen it, no problem. Okay, yeah, that looks nice. It's not particularly informative as is, but it will help us to complement the analysis that we do later on. So let's get back to the presentation, and then we'll see why that tree is so useful to us. So it is useful for a type of analysis called ecological diversity analysis, and ecological diversity analysis uses ecological diversity metrics. So the main idea is it's still fairly hard. We have all those different amplicons in each sample, right? But we kind of want to start maybe getting some descriptors of the ecosystem that each of those samples represent in kind of terms that we can kind of look at and actually interpret. And that is basically done with ecological diversity metrics. There's two different families of it: alpha diversity and beta diversity, whereas alpha diversity means ecological diversity within a sample, and beta diversity would be ecological differences between samples. So one acts on a single sample, and the other one only acts on pairs of samples, kind of comparing them to each other. So let's look at alpha diversity a little bit. The question is basically, in terms how diverse is a single sample? The most obvious thing you could do is, since we kind of have already that unique amplicon sequences, and we say that, oh, those kind of relate to individual organisms, we could look at each sample and just see how many unique ASVs are in that sample. And that metric is usually called richness. So how many taxa or unique ASVs do we observe in total? That can sometimes mask a few differences between samples. So like here's some example on top where we have kind of sequences coming from C categories like rectangle organism, triangle organism, or circle organism. So we have one that is, has kind of 3 different ones in here, but then the second sample here also has 3 different organisms. If we talk about richness, those 2 examples are exactly the same, right? They all include 3 different organisms. But the structure is different by far, right? We do see that in the first sample, they're all kind of equi-abundant. So they're very, the abundances are kind of very uniformly distributed. Whereas in the second sample, it's still dominated by that circle organism when we don't really have a lot of the other organisms. So we kind of maybe want to introduce that as well. If you would kind of only base it on abundances, then it would be called evenness. So how evenly are abundances distributed across taxa? So those 2 samples would have very different evenness. And then kind of what is used most commonly are kind of mixtures. So those are mixtures that take both into account. One is the Shannon index and one is the Simpson's index. So the Shannon index is probably the most commonly used measure of alpha diversity. It's a bit hard to interpret, because it's actually kind of derived from entropy or information theory measures. But one thing you can interpret the Shannon, it's basically a score that goes from 0 to almost infinitely high. Even though you don't see it infinitely high, you see something up to 8, maybe 0 to 8. But then a higher Shannon index means a higher level of being surprised if your system would be random and you just take out things. So if you take think of your ecological system as a random bag of organisms, and you grab into that bag and just see what you pull out every time. So for instance, in that kind of last case here, where we have a single organism, is basically no diversity present, right? The first time you would grab into that bag, pull out that circle organism here. The second time you pull out the circle organism, well, the third time you would not be surprised to see another circle organism, right? Because that's the only thing you have been pulling out of that bag or ecosystem up to now. Whereas in the first one, every time you grab into that ecosystem bag, you would pull out a different organism, always being fairly surprised at what you found. So basically that would mean that would get a really low Shannon index here, and the first case would get a really high Shannon index. And then the third case, well, even though there are 3 different organisms, it's kind of a mixture. So I might be surprised if I'm lucky enough to pull out one of the triangle or rectangle organisms here, but most of the time I still pull out circle organisms. So we'll get kind of an intermediate score on the Shannon scale. So that's kind of an interpretation of the Shannon index. The other thing that we might ask is, what if we want to compare compare alpha diversity? So for instance, maybe we have groups of samples that we want to compare to each other. Well, the nice thing about that, like for instance, the Shannon index, well, it's a single value for each sample. So you can use basically anything that would usually use if you want to compare a single measurement across samples, right? So you can use, for instance, a t-test or Mann-Whitney U-test, things like that will all still be working. So it's a bit more tractable since you don't have those complex things anymore, like, all of those Amplicon sequences, what with their unique sequences and counts, you kind of reduce it to a single number per sample that kind of quantifies that alpha diversity measure and can be compared, which is nice, but it's really hard to interpret what that actually means, and we will discuss that a little bit later. And the second measure is like better diversity, and that is more like, how different are 2 or more samples or donors or sites from one another. So basically, I have several samples and I want to kind of compare to each other. And, how would we do that on a global scale? Again, it kind of depends. So for instance, in the first example, that might look fairly similar, right? We don't only see similar organisms in both samples, but the abundances of the organisms are somewhat related to each other. At least we have 1 triangle in each sample, even though we do have like, different numbers of the circle and rectangle organisms here, but it's somewhat similar. We could also compare samples where we might have no overlap in organisms. So one sample doesn't even share a single organism with another sample, so that we would call really different. There is a bit of a catch here. And one thing is that even though the organisms are different, they might still be very related to one another, right? So you might see that, for instance, well, those don't really share an organism, like the strains, bacterial strains, for instance, are completely exclusive to each sample here, but then maybe we do see that the triangle and circle strain are extremely related to each other. So they're like evolutionary speaking, very similar. So should we take that into account or not? And then again, there's the question of whether we should consider the abundance or just like the taxa present. So for instance, if we only care about how many of the taxa are shared between 2 samples, that's often called an unweighted beta diversity measure. So just how many taxa of all the taxa are shared between the samples. So in that case, in the first sample, that would actually be weighted as extremely similar in that case, or perfectly similar, because it ignores abundance. It just looks at how many of the organisms are shared, and both samples include exactly the same organisms, so they're perfectly similar in that sense. Whereas the second sample here would be most different. But then there's also a weighted alternative, where we also see not only, do they share the same taxa, but do they also have the same abundance? And that's usually called a weighted, because basically the better diversity metric gets weighted by the abundance as well. And there's like different ones again. Like for instance, Bray-Curtis distance, which kind of checks kind of goes through all the taxas first that are shared between the samples and then starts comparing like the abundances to each other as well. And then we have like 2 distances here, which are both called Unifrac, which sounds kind of cryptic. So let's talk about that a little bit. Unifrac distances kind of resolves that issue that we had before when we were saying, well, we kind of want to take the phylogenetic similarity of those samples into account as well. And how do they do that? Well, they kind of do it with those trees that we just looked at. So that's why we kind of needed to generate them. And the main idea is kind of instead of just labeling the tips by the ASVs, the Amplicon sequencing variants, I can label them actually by the sample that particular sequence variant came from. And if I do that, I get kind of those bi-labeled trees here for every 2 combinations of samples. So I have those tips which kind of indicate from which samples they came from. And what I can basically start counting is I can go up this tree and kind of check how far can I actually go from any node before I get to an internal node that kind of crosses those sample boundaries. So for instance, in that case, this node is exclusive for that sample, and then the pink node is specific for the pink sample. I can go up 1 level, but then I already encounter my first node that kind of diverges, right? Where we have like 2 samples in here. Whereas, for instance, if we have a phylogenetic tree where basically phylogeny is perfectly aligned to the sample structure, basically all the sequences from a single sample are more similar to one another than they are from any other sequence from the other sample, then we get a tree like here on the right. And I can basically walk up that tree to the root node without ever encountering really a node that kind of diverges. The root node is the first one that actually groups samples from, ASVs from different samples. So that length of how far I can walk up that tree, that's kind of what Unifrac uses to quantify how similar, phylogenetically, maybe ASVs or samples are. And basically just counts the length of those kind of divergent or unshared branches and just divides it with the total branch length of the tree. And then we kind of know that in the first sample, that's a fairly small sample, very small distance, so that means small distance means very similar. In the second example, that would be basically a distance of 1. It means they're very different. And the weighted Unifrac, the only thing it does is that it also weighs those branch lengths by the abundance. But that's like a good way to also integrate that phylogenetic information and then kind of map that. So that's what we needed to generate that tree, because that gives us also a better estimate when we want to calculate better diversity later on. And why do we actually care about beta diversity? What can we do with it? Because beta diversity is still a very complex data type, right? So for every pair of samples, you will kind of get that measure that just tells you how similar they were, right? So that's kind of still hard to quantify and hard to compare. So what we kind of can do with it is something we actually do a lot, because there is, it's a distance matrix between pairs of things. There is actually, a very common data types that we all kind of deal with almost on a daily basis, which are just, distances on just geographic distances, right? Whenever you want to look where to go, you kind of, take that into account, because you kind of need to know how long it would take you to go somewhere. So here's an example. If we, for instance, would take 3 cities and we have that distance matrix, how do we represent it commonly? Well, we draw a map, right? That's what we look at if we want to intuitively grasp distances. And it actually, there's an extra step to it, because those distances, if we take it in real physical distance, there's a lot of aspects to it, right? It's not only what's the, direct airline between 2 cities, but it might be, what streets can I take? That might actually be longer, the street distance. There might be a height difference, just because one of the cities is maybe very high up the mountain and another one isn't. So it's kind of a multi-dimensional distance, it's at least acting in 3 spatial dimensions, but there's also other dimensions, maybe as roads and so on to it. But what I can do is I can reduce that maybe to 2 dimensions, which is similarly a map. But the same as a normal geographical map, there's several projections that we can do, and that might still give us information, right? So we do know that, for instance, for the world maps, there's several projections that kind of try to do it. None of them are perfect, because exactly the real world isn't a 2-dimensional system, right? But yeah, we do use those simpler projections to kind of get to that step and to kind of simplify it for us. So really similar, that's exactly something we can do. And that's actually a perfect example of a principal coordinate analysis. So a principal coordinate analysis is exactly that. Take a really complex distance matrix and then represent it in low dimensions, usually 2 dimensions or 3. that kind of are still very consistent with the distances. So basically, if we look at the map of those cities, you know, the distances we kind of perceive from this map line up very well with kind of that distance maps that we had initially, which kind of means it's a consistent map. And that's what we're looking for. We're looking for a representation which is very good and usually the best under certain assumptions and kind of like preserving those original distances in that like kind of reduced space. But similar to a map, there are certain things that are not really conserved. So I can take the world map and I can just turn it upside down and all the distances between cities will still be fine, right? And that's the same. So every time we run this principal coordinate analysis, we might see like rotated versions of the same information, but the distances of each point will always be consistent. And then, and maybe like the last discussion point for that first part, is like, how do we do statistics on that? Because that's way more complicated, right? We kind of looked through that better diversity metrics and distances of things, and that's kind of complex to do. So what we can do is like on that map, we could kind of look, is there like a different centroids between groups of different samples? But if we want to do real statistics, it becomes kind of iffy because we have that very high dimensional thing still, the better diversity measures. And we want to maybe fit it into, you know, normal regression models or something. So how do we do that? Well, there's certain levels to it. You might all have heard of analysis of variance models and ANOVA models. So basically, there's a question is, you know, does the particular grouping that we imply in our samples kind of explain a lot of the variance in the data? So do we see, for instance, that like a lot of the variance that we observe between samples is kind of like related to, for instance, the geographic regions the samples were sampled from or something like that. But then if we do that for better diversity, it becomes more complicated because it's not a single number that we regress against. It's like actually what is called a multivariate ANOVA, because we get, you know, for every single sample, we have those comparisons with any other sample, which would still be fine. But then microbiome data is a bit tricky for several reasons. So kind of, you know, using all the common methods that we would know there would usually not, you know, map that variability well. There's a way to do it in a way without assuming certain distributions, which is called PERMANOVA, which is permuted multivariate analysis of variance. It's quite a mouthful, so we usually take a shorter acronym for it. And the main idea is basically, we have our better diversity measure. We have the real groupings of our samples. For instance, we know which geographic regions the samples came from or which communities the same samples came from. What we can do is we can scramble that assignment like on purpose randomly. So we kind of take that and scramble the assignment of the geographic region the sample came from, for instance. We can do that quite a bit of times, depending on, you know, how many samples we have. That explodes quite quickly into possible permutations we can do. Usually we cut it off at the maximum number of permutations, maybe 10,000 scrambled versions. And for each of them, you can also run like a regression model, just kind of look at, you know, what you get out of it. So you kind of can fit the model and you can extract something which is usually goodness of fit, like an F value. An F value would kind of tell you like how much more variance are you kind of explaining, more or less, like it's related to that at least. And we have a real F value that comes from our correct assignment of geographic regions to samples. And we have those scrambled F values that come from a permuted version. And we can compare them basically, right? So that scrambled F values are kind of our background signal. Those are the F values that we would expect if our group is basically completely kind of randomly assigned to the sample. So it's kind of non-informative. And we can kind of compare that our real F value, and that can even be used to calculate a P value. And it's nice because we kind of keep a lot of the structure of the data because still using exactly the same better diversity matrix for each of those regressions. We're still using kind of the same like prevalence of like groups. So you would, you know, if you have 100 samples from one geographic region, 10 from another, that's still conserved even in the scrambled regressions, but the assignments are just scrambled. So you conserve a lot of the structure of it, and that tends to work out really well, like to kind of test. And you can use that to kind of see, oh, is there like some kind of a, you know, a notable difference between the better diversity metrics if we start like comparing certain groupings of the data. And that can also be used to correct for certain things and see to pull that information out. And with that, I would actually think, since that has been quite a bit of talking from my side, before we actually go and really run the diversity analysis in the notebook, this is a good point to take a short break. We are kind of very close to what we had original in the calendar, so we can kind of go into the break a little bit earlier, and maybe I can also add in a little bit of a quick Q&A session if some of the TAs, this was like a topic that popped up repeatedly on the Slack channel. I know that I went over certain things pretty fast to kind of keep in time. So yeah, if there's some unclarities, now is a good time to ask.
There were some questions about what does it mean to read count, read depth, coverage. So maybe I think know as 6US, so maybe some discussion of that.
Yeah, let's go back to those slides. So like just to rephrase the question, because I'm not sure what could be here, there were some questions about differences between things like read counts, read depths, and coverage. So a read, like I said, is that single thing that a sequencer generates, which is a single readout from that sequence. But we have many copies of every single sequence in the sample, like because since we amplify it, we get like a massive amount of copies on any of those sequences, and the sequencer might actually also sequence through certain copies a lot more. So basically that's the example here. If I have a single sequence and it generates a lot of reads, this number is called the read count. So how many of the reads assigned to that sequence. And then later, if we do the grouping in data 2, it might actually, collapse even more, right? Because we see that, a lot of reads actually came from the same Amplicon sequencing variant. And the read count is just that. It just means how many reads in the original sample were actually stemming from that one Amplicon sequence. So it might tell you that this Amplicon 1 had like 1,000 reads assigned to it, and Amplicon 2 only had 100 reads assigned to it, and so on. So that would be reads and read counts. The coverage usually describes, that's more informative probably for metagenomics or shotgun metagenomics. The coverage is kind of that profile that you see here. So since you randomly fragment the genome, you might see that certain areas of the genome actually get more reads assigned to it than other regions, just randomly. You might just have like a bit more sequencing on one part of the genome than on another. There's also other reasons that can happen. But like kind of the coverage would be, if you map that out across the genome, that profile, like how many reads map to each position of the genome, you get kind of like, a plot that kind of looks like that, which has like valleys and peaks, and that's often called the coverage profile. So it tells you how often was each of the nucleotides in the genome actually hit by sequencing, and that might be, fairly high in some regions and lower in others. If you collapse that, if you just take, for the full genome, on average, how often was every single nucleotide hit on just in the mean value, which is the single number, then you get the read depths. So that tells you like how deeply you sequence, and that's often expressed as something at like 10x or 1x. So 10x would mean every single nucleotide in the original genome was at least hit 10 times by sequencing reads. So like you kind of have almost a guarantee that every part of the genome was kind of covered pretty well. For metagenomics, you see massive variations there, because it depends heavily on how abundant that organism is. Like very abundant organisms will have massive read depths. So you might see like 10, 20, 100x. And then you will have a whole bunch of organisms that are low abundance that will have like below 1x depths. So that kind of means that like it's very unlikely that every position in the genome was actually covered by reads. Yeah.
So there is a question about data interests about parameters for when you filter out samples, or what's the minimum read depth. How do you make these choices?
Yeah. So the question was like, how do you choose where to kind of remove, which samples to remove because the quality metrics aren't really great, for instance, or what are those? So the 10,000 is kind of based on, how do we actually get to making good choices here? That's mostly based on experience. So basically what you can do is you can take a really deeply sequenced samples and then artificially subsample it, right? Like if I have 1 million reads, I can say, okay, what if I take 100,000 random reads from that sample and then 10,000? And I can build a kind of upper curve and I can see when I start losing basically the information from the full sample. And that's like often done with very high sequencing depths, but that data is what's used to guide those rules, like as, oh, if you sequence higher than 10,000 reads, again, that's often, still influenced by representation issues. Like I said, a lot of those like kind of rules of sums are generated by the data that's available, which is again overrepresented. So that's something to consider here, but yeah, that's kind of where that was based on. So we people have sequenced 16S samples with really high depths and a lot of reads, like total reads per sample. And then if they kind of do that artificial subsampling, they see if you, at 10,000 reads, you still maintain a lot of the diversity metrics don't change that much, for instance. And the abundances you get for certain tags are kind of maintained. In terms of quality, that really depends. So if you have heterogeneity and the length of your reads, you probably want to pick something that still maintains the majority of your reads. So you can also do that data 2 pipelines that we did repeatedly changing the parameters. Might be a bit lengthy, but might be good if you do it the first time to establish like a baseline. So for instance, you can truncate at different levels or like be more strict with your quality score cutoffs. And then you can always look at those statistics that we looked at, how many reads were maintained through the pipeline. Because if you're too strict, you might lose a lot of information. So if you lose 50% of the reads from your sample, is that still representative in the end? Probably not. So you kind of want to maintain it above that level. But those are good views. And for qualities, really looking at the quality profile usually gets you a good way. Like also looking at if there's really wonky signals, sometimes you might see your quality profile look really odd in a certain region. So you might want to exclude that from analysis and so on. Yep, those were great questions. And yeah, we'll be looking at running our ecological diversity analysis after the break. But yeah, right now we can all take a short breather, get a beverage or snack, and yeah, recharge batteries a little bit before we go on. Thanks so much. Okay, everybody, welcome back to the second part of our QIIME 2 course today. Yeah, we went through a lot of different concepts already in the first part. And the last thing we talked about were the ecological diversity metrics and, how to compare them and calculate them. So like now is a good point to actually, go back to the notebook and run them. So we can switch to the notebook again, which hopefully should still be there for you. If not, you can also just rerun all the previous cells. And one good thing is that for QIIME 2, we have basically a plugin that can actually generate a lot of different diversity metrics at once and generate a lot of different artifacts, kind of similar to the Data2 input. So the one that we were looking at is the core metrics phylogenetic diversity plugin action. And as an input, it basically takes the abundance table generated by Data2. So the abundance table in Data2 basically had the counts for each individual ASV sequence. And that we will be using for all the comparisons. We do input also the phylogenetic tree that we generated from the multiple sequence alignment. So we can use like metrics like Unifrac. And we do something called, like, we specify sampling depths, which is a bit odd. So why would we want to do that? And it has to do something a little bit with normalization. So what we did see initially is that across each samples, we had fairly different numbers of total number of reads. But the original abundance counting just will, count reads for each ASV. But that creates a little bit of a bias there because, if you compare a sample that maybe had 10,000 total reads to another sample that had 100,000 total reads, then you might observe that most of the ASVs in the sample with 100,000 total reads is more abundant, simply because they still have the same relative abundance, but they kind of have a higher total read depth in the sample or higher total read count. And specifically because there isn't really much information contained in that. So you might maybe say, oh, well, one sample has 100,000 reads. Maybe there was more DNA in the sample, or there was like more bacteria in the sample total, but that's not really true because the one thing we do before sequencing is usually normalize the amount of DNA. So for every sample, we put in a constant amount of DNA. And all this difference in different reads across samples is actually just an artifact of the sequencing. So it's a very microscopic process that often can happen that certain probes just attach better to the sequences than others, and you get those differences in total read counts of current sample, which is not really associated with any real signal. So what we can do is basically can get around that is we can subsample each sample to, a small amount of reads, which is kind of maybe the minimum number of reads that we had for every sample. So if we have a sample with 100,000 reads, we just take 10,000 random of reads of those total set of 100,000, and we pull that out and do the analysis with that. That is often called rarefaction. So it kind of subsamples it all so that we have the same total number of reads in each sample. There, for certain statistical analysis, that might not be the best strategy. It works well for like visualizations, like PCoAs and things, but there might be also other strategies, and we'll look at different methods to normalize as well later on. But that's something that we will do here. And then we also pass in a metadata file now, which is the first time we actually do that. So a metadata file, like I said, includes information about the samples. So in our case, it maps the sample to the communities that sample came from and gives some information about where those communities usually get their food from. And that we will use so we can kind of, for instance, compare across the geographic regions that we sampled from in our later visualizations. And then we generate an output directory for it. Again, that's the same strategy as we did for earlier. So if one of the actions generates a lot of different artifacts, we just specify a directory and it will just save them all in there. So we can run that. It will not take too long, even though it does go through quite a lot of calculations, like, generating all those different metrics, the alpha diversity, beta diversity, and so on. And the first thing we could do, for instance, after that is and you see all the artifacts that are generated by it. The first thing we could do is, for instance, try to, look at the Shannon diversity across those. So basically, we could take the alpha diversity, which is a single value for each sample. And now we could say, like, for those samples coming from different communities, how do they compare? And we can look at that by using the alpha group significance action from the diversity plugin, which essentially runs the test for that. And I think it runs the Mann-Whitney test by default. And we'll also create like a box plot of the Shannon diversity across those groups. So same strategy as before. This was saved into diversity and then alpha groups QCV. So we will look for that folder. We might have to again update it to actually see it. Alpha groups QCV, that's the one I'm going to download. And then look at it again with the visualizer from QIIME 2. So what we see is, well, the major signal here is that we see about all communities, the alpha diversity is actually pretty high. So above 4 or something is, you could say that's high alpha diversity for the human gut microbiome sample when mapping to the ASV level. And by default, it actually integrated also the metadata table. So those were all columns in the metadata table that we provided. So that's a file that you can generate yourself. And you have the other ones as well, which in this case, they're actually, you know, kind of all different between the groups. So we can't actually distinguish like, you know, if some of them are driving more or less of that, because like we do see that they're all fairly different. But what we do see that here and that the samples from the Hatsa community have an exceptionally high alpha diversity, which is also owed to many factors, but also the very complex diet. And then we also see, you know, that the other ones also still have fairly high alpha diversity. One thing is actually what is complicated here is the interpretation aspect of it. It's actually hard to say what that means. So it is, even though it's a property of the ecological system, it isn't necessarily connected to any function. So usually you'd actually see more correlations if you would look within a community. So if within a community, you try to correlate alpha diversity of the gut microbiome to certain phenotypic measurements, you might see a signal, but across communities, that's often mostly driven by geographic differences and like just different lifestyles and so on. So that's really hard to tease apart in that case, but we do see that there's definitely patterns to it here. And then it might also run a statistical test for it. In this case, it's 3 different groups. So that actually you can kind of see the pairwise comparisons, but you can also see like, then the cross-group all is pairwise is just the Mann-Whitney actually. And it will also correct for multiple testing if you want to see if, you know, all of them are. In our case, they are fairly, I mean, they're visually very different values, and it also assigns a statistical significance to that, saying that, you know, they seem to be statistically different alpha diversity measures here. Okay, and I can go back to the original one. That's the first thing. So you could also look at the PERMANOVA analysis that we talked about, which was that very, you know, funky method of trying to scramble the data and then extracting goodness of fit values and getting p-values just from that. So for instance, you could kind of see if, again, like even if you look at better diversity, so just looking at, you know, on an ecological scale, are the samples, is some gut microbiome composition here and that very better diversity scale like different between different ethnic groups in that example. And if we look at that, you can kind of do the same thing, download that PERMANOVA QCV visualization, look at it again, and this time actually just gives us a table and it tells us how much of the variance is explained. So in this case, it tells us in that better diversity metric, 61% of the total variance is explained just by differences between the different ethnic groups. So that's really a large fraction. The majority of the variance is actually just explained by that, which is, you know, not surprising, having communities from very different locations on the globe. So that's not something that should surprise us here. But yeah, it's kind of like shown here, like, and it also assigns a significant p-value to it. So we do see that, you know, the gut microbiome, like on a very broad ecological comparison scale, there are very different compositions that we see here. And a large fraction of the variance is explained by the grouping into different communities. And it also generates a lot of visualizations by default. So if you look in the diversity folder and you look for files that end in QCV, you see a bunch of them that we didn't really sign with a separate comments that were generated in the beginning as well. And you see that they're usually prefixed with the name of the distance So we might remember that Bray Curtis was a weighted beta diversity measure that did not take phylogenetic distance into account. We do know that the Unifrac versions do take that into account, so maybe we want to look at one of those. And one thing we might be looking at is a weighted Unifrac. So that was the version that took phylogenetic similarity into account, but also weighted by abundance differences, so kind of incorporates all the information. We could have a look at this one as well, and that will look pretty fancy. I'll go back to the viewer again and pull that up here. And feel free to also look at this in way more detail on your own time. So now we kind of see that projection. By default, it actually projects in 3 dimensions, not in 2, but it does align it along the first 2. So it actually looks almost like a 2D map, but you can drag it around with your mouse to actually also see the 3rd axis if you wanted to. So by default, for instance, you can also only look at the first 2. We see that the first axis, they're also annotated by the amount of variance in the data explained by a single axis only. And you see that a lot of it is explained by the first axis and then a larger fraction by the second. So the sum of both is already pretty high. So we do capture a lot of the variance in those distances. And you do also have that chance here to color those initial points by the data from your metadata. So if you color them by the ethnic group, we actually see, a little bit of a surprising signal here, but that actually, like, there's one sample from the Japan groups that actually clusters a little bit with Semifa, which have like a large diversity as well here. But in general, we see a tendency that, samples from a particular community tend to cluster with samples from that community, which again is to the fact that they're from distinct geographic regions, but we do see that there's also some finer mapping here, and that has also a lot, again, to do with, lifestyle and so on, and the way we quantify that. Okay, so that's how we looked at that. Interpretation is a little bit harder here. Still, we don't really know what, we can see that there may be differences, but it's like another challenge to really see what that means and how do we interpret it. And we also have to see that a lot of, the social aspects of that should be considered as well. So, and then that gives us like a very global description of the different samples, but we now might be getting to the question, who's really there? So who's at the party, who's present in the microbiome, and try to quantify that as well. So what organisms are present in our sample? So how would we do that? So yeah, maybe you can go in the Slack chat if you have some ideas how you would usually do that. So the information we have up to now is just what we got from our data 2 analysis. So we do have the sequences of the individual amplicons, but we cannot map that directly to names. So what would be a strategy there? So what would you do to go from a sequence of an amplicon to an organism's names, essentially? For instance, to be able to tell, oh, that sequence came from E. coli, for instance, or from Bacteroides, and so on. And there might be some suggestions. And while those pour in, let's talk a little bit about taxonomic ranks. So usually how that works is that in bacteria, it's actually quite complicated to really, group them into distinct clades like that. But in really general terms, what we have is usually we have in taxonomy. Taxonomy is kind of like a hierarchical ordering of namings, where names are usually contained in larger groups again, and those groups are contained in other groups. And a good example of that is like what we usually know when we assign, for instance, species names and genus names, right? A species name is a very specific name. It's not the most specific you can go. The most specific is usually a strain, which means like a unique genome. But then we do know that, most individuals in a species group usually have very low genomic divergence. So they're pretty similar on a genomic scale. And then if we go up the taxonomic ranks, we kind of get less and less similar groups. So the genus is a bit more heterogeneous than the family again. So we can have those groups, and we use that basically to assign names in a taxonomic system. For amplicon sequencing data, honestly, the lowest of those ranks that we can reliably detect is the genus level. Like within a genus or within a species, the V4 region just doesn't differ enough for instance, that we could really detect those reliability. Sometimes you can assign a species to like a few of the ASVs, but that fraction is usually really low. So you might only represent 10% of the total reads in your sample, whereas on the genus level, you get the majority will get a genus assignment. So maybe one of the ideas that was mentioned in the chat is that essentially we could look for, what is the actual sequence of the 16S amplicon in a reference genome, right? Like when that organism was sequenced and really well assembled and curated, like we could take the known sequence and compare it to the ones we actually found in our sample, and then say if they're very close, maybe that organism is present. That seems intuitive, right? But it doesn't always work well in practice. And why? Well, the reason is mostly because those reference databases might not really, correspond to our organism. So it might actually not have been observed, but there's also, there might have been some divergence. So even though our reference genome was maybe for the lab strain of E. coli, what we actually have in our samples is an E. coli strain that was always living in the human gut. So it's not really the same organism that was, for instance, sequenced in the lab. But we can still kind of use those database of known genes to kind of come up maybe with a slightly smarter way. So instead of saying I want a perfect match to a sequence that I've seen in a reference database, I can ask are there characteristics of that reference database sequence. And one idea would be, for instance, that a lot of bacteria adapt, for instance, their usage of codons for a particular environment. So as we know, is that the genetic code is degenerate, meaning that like different codons of nucleotides can still encode for the same amino acid. And that can kind of create like a strategy. For instance, really basically, if you had that idea, you could base it on that. You could, for instance, ask, okay, is the codon usage, for instance, in the 16S variable region different or very specific to specific species? And we can actually generalize that even more. You can just ask, well, if I just look at short subfragments, what does that look like? And that strategy is kind of what's used in a multinomial naive base. That sounds super elaborate, but it's actually just that basic idea. So you take a longer sequence and you just subdivided in all possible smaller subsequences, and those are often called QIIMEs, for like a small substring of the sequence with length k. So for instance, a 2-mer would be all subsequences with 2 nucleotides in it, and a 3-mer would essentially be a codon, right? Like 3 nucleotides. And that's kind of shown here as an example. So you can kind of slide over the sequence and just count how often each of those subsequences appear. So for instance, here in that longer sequence, we see we have 1 ACG, 2 CGCs, and 1 GCG. And it turns out that if you take those QIIMEs and you make a profile out of it, so you kind of say, oh, how likely is that QIIME to appear in a certain 16S gene from a certain species, for instance, you see that they're very specific. And it's like a little bit more generalizable than just looking at the exact sequence match, because it does allow still for that sequence to have diverged a little bit, but it takes into account that, as evolution goes on, or as, strains diverge, maybe they usually don't tend to do it too much. So we kind of observe a lot of that profile is kind of maintained. So what we do from a reference database is we build up that reference model where we kind of have, for instance, 2 different taxa in the database. I might take into account that a taxon is very overrepresented in the database. Maybe I see that, I have 15 16S sequences from E. coli, but only 1 from Bacteroides. Maybe that's something I want to take into account. And then I can kind of build up the frequency or the probability of observing any of those QIIMEs in that particular sequence. So in the first case, we kind of know that we had like, 1 in 4 chance for an ACG. We have a 50% chance for a CGC, actually, sorry, that's kind of turned around here. And then the GCG would be a force as well. But essentially, you can kind of count those examples, and you can do that for each taxon. You get those profiles, the probability distributions of QIIMEs. But why is that in any way informative? That just tells us how likely it would be to observe a certain QIIME from a different taxon, but we actually want to do the opposite, right? We want to put in a query sequence and then know what's the probability that that query sequence came from a specific taxon. And that's where the Bayes part in that method comes from. Bayes is just a really smart way to turn those things around. So if I want to know what the probability is that, that a particular query came from a taxon, or the probability that I was actually observing taxon 1 given that particular query, then the Bayes theorem says that, oh, you can turn that around. Like as long as you know the probability of a certain query given a certain taxon. And that's exactly our reference model. It tells us that, right? Because it gives us a rule how to calculate that probability by basically going through those individual ones. Because if I have my full sequence, it would be exactly that. What's the probability of the query given its taxon 1? And that's calculated basically what's the probability of just observing the taxon, even though methods might differ here and into taking that into account, or just saying that the probability of observing any taxon is uniform. And then that would be the probability of observing 1 ACG, right? And observing 2 CGCs and observing 1 GCG, because those were exactly the k-mers in the sequence. And an and in probability usually means multiplication. So you can just multiply those out, and you get a total probability just calculated from the QT part. Then there is that probability of T part here still in the formula, and also the probability of observing a query. Usually we just say we don't really know the probability of observing a certain sequence, but we will just assume that they're all kind of equally probable. Then we can just kind of not consider this part that much, like because we kind of know that still if the part up here is kind of maximized, then it doesn't really matter what happens in the denominator. So that is kind of what we do. There's also like more general methods. You can use any machine learning methods based on k-mers because they just give you a profile and you could classify on that. But the multinomial naive Bayes function works really well. So it provides a bit better generalization because instead of matching exact sequences or something, it kind of takes those parts of the this prevalence of certain k-mers into account, which is a bit more stable in most cases. It also allows you, for instance, to classify sequences that you might not have been observed in a reference database. It might, for instance, tell you, oh, well, I'm pretty sure that sequence is coming from this particular order, even though I can't really assign a genus. So you can kind of do things like that, even with sequences that haven't been observed. So without further ado, let's just go and actually run the taxonomy classification. The way that works is that you usually have already like a classifier that has been trained on a reference database. There are many of those available. The largest one is probably the Silver database, which is a reference database that only contains 16S genes. And that contains a lot of organisms, because for many organisms, you might not have a full reference genome, but you might have this 16S sequence. So, but we will be using a bit more like a reduced data set here, which is still working really well for most cases, and also will interface better with what we do tomorrow, which is the NCBI RefSeq database, which is basically just the database of any organism that has like a full reference genome in the NCBI database that's well curated. And it will also work better in the Google Colab setup. So basically that's the reference model. And then we just apply the Bayes theorem essentially to find out what is the taxon with the highest probability given those particular input ASV sequence. And that's what we get. So for each ASV, we get like a taxonomic assignment attached to it. And that will take a little bit, but not too long. It's a fairly straightforward job. We use the classify scikit-learn action from the feature classifier plugin. Like I said, what it now needs is the actual ASV sequences, and Data2 did generate those, and they're contained in artifact representative sequences. We use our NCBI RefSeq. That was kind of trimmed at the genus level, like I said, because that's the finest level you can reliably identify, and was actually trained also only on that subsequence that corresponds to our amplified regions. So you kind of see the primer pair that we used here appearing again. So that gives a little bit of higher sensitivity than, for instance, using the full 16S gene, because that would also take into account the V1 regions, also conserved regions, but we actually only do it on that particular fragment. So then we get like a taxonomic assignment. And that kind of for the first time allows us to actually look at like, you know, not only which particular bacterial taxa were present in the individual samples, but also how much of each of those taxa were present. And we do that by basically combining that information of the assignments of taxa with the abundances we get from Data2 and generating a visualization, which is a bar plot. And that is in the taxa plugin, and it's a bar plot action. It takes as an input two things: the abundance table, we got individual for the ASVs, and then also the taxa assignments, so the taxonomic assignments for the ASVs that we now generated. Again, we put in our metadata as well that kind of annotates the samples a little bit, and we get a visualization out, which is, well, we will see how that looks like. We might have to update it. It should appear in the top level of the materials directory. So you should see that taxa bar plot file here. We can download that. And same procedure as before. Oops, clicking at the wrong thing, but then pulling it up here. So now we kind of see for each of our samples, we see it, and by default, it takes like the highest taxonomic level. In here won't necessarily tell you what's like the kingdom and so on. You can kind of infer it because it prefixes it in front of the name. So here we see the kingdom. Usually the V4 region is very specific to bacteria. It doesn't really do a great job of detecting archaea, but there is a few archaea that can be identified based on that if they're present in the database. We see that like for one of the samples from the MEFA, we do see like a certain relative abundance of archaea, even though there's probably more in other samples as well that just wasn't detected here because it's not really great in getting archaea out. But we can also reduce that. For instance, we could look at the phylum level, which is usually, you know, a good indicator initially to look at because you kind of know that those are not too many groups, so we can still color them well on a bar plot. And you can also sort samples by, for instance, ethnic groups that were sampled from. So we kind of get them a bit nicely grouped. And we do see, again, that also here we see quite some differences. And actually, so in those communities, you might see that, you know, a lot of things that you might have heard, maybe from, you know, the typical European and North American population doesn't necessarily extrapolate well to like those samples from communities that are not really represented well in the existing literature. For instance, you do see actually large abundances of Firmicutes in the Hadza, but the Hadza still live on a mostly very plant-based diet. Meat isn't really that often consumed in the Hadza communities, which is, you know, like often you hear rules like, oh, high Firmicutes is for high meat consumption or something, but it does tell us there's many ways to assemble an ecological system or an ecosystem, like in the human gut, and a lot of the rules don't really necessarily extrapolate well into communities that are understudied. We do see that those, even on the phylum level, we do see quite some differences across the different populations, again, due to many reasons. So there's definitely quite some heterogeneity going on here. And you might map that even to finer levels. You could look, for instance, at the finest levels that we have here is the genus level. You can kind of see that, for instance, like some of the Chepang and MEFA samples have a lot of high Prevotella abundances, which is kind of almost dominating here the microbiome, whereas like the Hadza samples, you see really like consistent with that alpha diversity measure we observe. We also see that, you know, they have a huge variability of different bacterial genera that live in their guts. So that gives us like an information who is there. One thing you might want to compare is you might want to, you know, say, okay, what if I take my abundances that were mapped on the Amplicon sequencing variant level and kind of want to collapse it maybe on like a higher taxonomic rank. So that's kind of what we saw here with the relative abundances in the bar plot. For instance, if I collapse on the phylum level, I see that particular percentage of Firmicutes, for instance. You can also do that and generate that data. And one of the last things we might want to do is kind of, you know, also show an example how you get that data out. So usually a lot of those taxonomic abundances or like on different ranks is something that we might want to use for different downstream analysis. Maybe there is nothing available in QIIME 2 that does a particular analysis that you want to run. Maybe you want to run a particular statistical model or, you know, use a different programming language. Maybe you have like some R scripts to analyze it and want to do that. And that is something that we could definitely do, and we will show some examples. So here's like a few examples here. So we can kind of do like a collapsing and kind of take the abundance data that we had from the other two and kind of collapse it on the genus level, for instance. Actually, I see that it's using the treasure chest data here. You could also remove the treasure chest part. It will give exactly the same output. It's just, you could also run it just like that. Just the taxa and the other two tables that you generated in your run. And it will generate a new artifact, which are now the abundances just collapse on the genomes level. That actually doesn't necessarily, it doesn't do any rarefaction or things like that, or calculate relative abundances. So it still has original read counts, which might be good if, you know, your particular statistical methods actually wants that data input. And then we'd also do an example here, which is like a two-step process that actually pulls out the actual data. So that's still an artifact. And I did tell you it's like basically a zip file, so you could go and rename it, but there's also tools to directly export it. So one thing we can do is we can go into can use a plugin called tools again, which we used to initially import the data. It also has an export function, but it basically goes into an artifact and pulls out the actual data. So we can run that. And then the first thing that would do is only, you know, pull out the particular data format that is here, which looks a bit odd in our case. So what we will see is, if we look at that in our exported folder now, is that it generated a file called .biome, which is nothing we would be able to look at. It's like kind of a very compressed version of those very large abundance tables, but we can kind of add in a tool, like basically there is a tool that comes with QIIME 2, which is called Biome, and that actually can convert that table in that compressed format to something you kind of know, and we will output it to a tab separated file, which is just, you know, something we can read with almost any other tool, and that's what we did here. So we also have that genus TSV table. You could kind of look at it, and kind of what it does is it now combines those information, so you kind of get the taxonomic information down to the genus level, and then you get the read counts in every single sample. So you could say that, you know, in the first Chepang sample, we had 3,697 reads that were mapped to Prevotella genus, for instance. So that you can actually plug in into any analysis that you wish, right? Because basically any programming language has a package to read TSV files. So it's like a very common format. And without like really understanding everything that's going on in the next fragment of code, I will walk you through it, but you don't really need to be like a total expert in Python or what packages to use. It's just to give you an example, like how you can use that data and kind of use it without kind of maybe using QIIME specifically. So that's actually a bunch of Python code here. I don't really have to prefix it with the exclamation mark because by default Jupyter just executes Python code. So I use like a bunch of packages here. Maybe the important ones here are pandas in this case, which is just a package that can read basically data frame format data from a lot of different formats, including TSV files. We have Seaborn, which is a plotting library to do a lot of different visualizations. So we kind of use that pandas here to, for instance, first read the table, which gives us like a data frame. And we can do a lot of stuff with that. There's a lot of helper functions, pandas. One thing is we could, for instance, split this kind of longer string. So if you remember that what it looked like, we have all those assignments here down from the kingdom level down to the genus level. So what we could kind of do is, if we want to reduce that a little bit, let's assume we only want the genus level. So we only want, for instance, the Prevotella part here. So we could kind of split it on the semicolons and also strip away the leading genus identifier, the G underscores, because then we know we already extracted the genus. That's kind of what the code here does. We can also remove ASV sequence that did not have a genus assignment. Like I said, those databases can generalize, but they might not be able to map to really low ranks. So if they're very low, not a lot of representatives of the specific genus in the database, or the particular sequence that you found isn't really represented well by those, then you might actually not see an assignment on the genus level. So we kind of remove those as well that have like an empty genus entry. So that either start with 2 underscores, have like G or 2 underscores. Those usually mean there's no assigned genus. We can, for instance, take 50 random genera from that set. And just because, you know, we want to be super elaborate, like instead of, for instance, using a rarefaction, we can use a different form of a normalization method, which is called a centered log ratio transform. It sounds super complex, but it essentially means is you log transform the data, and then you subtract the mean from the log of the data. What that does is basically it also kind of takes out a little bit that library size, so the amount of different reads across the samples, because like you could assume that this is just a random thing that like makes everything more abundant in a sample with more reads, and that will be basically removed by that technique. There's a lot of like there's a lot of technical or theoretical region why that is a good transform. It's also really good for things that sum up to a particular sum. So you usually know that all the relative abundances sum up to 100%. It kind of removes like spurious correlations a little bit that can appear because of that. But yeah, it's a different way to normalize it just. And you could use any other methods that you see fit here. Just an example how you transform the data into something that is now, a log transformed abundance minus the mean log transformed abundance. And it's applied to each single sample here. So essentially what that new normalized data means is if we see a value of 0, it means that's basically as abundant as the average thing in that sample. If it would be negative, it means that particular genus is less abundant than the average. If it would be positive, it means it's more abundant than the average. So it's kind of still has that information of what is like high and low abundance in the encoded, but kind of normalized. So you can actually compare a bit better between different samples. And then we can use like a really fancy heat map function from the Seaborn thing that kind of like draws like an informative heat map that shows us in each sample which are those 50 genera and how abundant are they at the same time. And like if we run that, it will create that particular custom visualization for you. And you could like tune that with different things. So basically what it will tell you now is, and as here like the ID of the particular genus. And then here you kind of have the different samples. So you can look at a particular genus in a sample and we'll kind of color them by the abundance, going from very dark towards kind of like a high yellowish orange color for very abundant things. So for instance, here in that sample, we see that Fecalibacterium is pretty abundant in all of those samples, but with quite some differences across different samples, right? And you do kind of, so that plot will look very different for each of you because essentially it creates a random subset, which will, come out as something different for all of you. So you will look, every one of you will have a very custom plot, their own personal list of genera that they're kind of analyzing here. But what you will all see is that there are always those patterns of some of those genera kind of present in all of the samples. So they're very ubiquitous, but then you will see some genera that are very specific, sometimes even for a single sample. So they only appear there in really high abundances. You will see that some of them appear in a particular group. So in a particular ethnic group, you will see that genus pop up with a slightly higher abundance. And then some of them might, have like kind of like a hybrid pattern, which appear maybe in two groups, but not in the other, and things so on. So you can kind of extract some of those patterns from there. And you kind of see that there's really a lot of heterogeneity in terms of like, how ubiquitous a certain genus appears. We do know that there's like a few of the taxa that probably appear in most human microbiomes, even though it's something we can't really say with certainty yet, because honestly, we haven't seen enough of the human microbiome on a global scale yet. But we definitely see that this is also because of those patterns that are very specific to specific ethnic groups, that can also sometimes create like certain misrepresentations of things. Because if I kind of base all my research on a particular genus that only appears, for instance, in people from the US, then this is obviously not going to be particularly useful for communities where they don't even have that particular taxon in their gut at all. So, so like that's kind of an argument to look at that more. There is a caveat to that here, though. The fact that we can assign a name to an ASV doesn't really mean it's the same organism. And that's something I want you to look at in that exercise section here that you can do on your own time. Essentially, since we now have the taxonomic assignment, we can come back to the kind of the beginning where we had that phylogenetic tree of the original Amplicon variances. And you will be able now to annotate that tree with those annotated genera, for instance, and kind of see, okay, if we look at the Amplicons extracted from that genus, are they really like very similar? Do we see that, they appear across all populations or not? And you would see there's quite some signals there. And yeah, I would like you to go through that a little bit, maybe reflect on that a little bit. What does that mean? What does it mean if we have high divergence? between different amplicons, so between different organisms in the same class of genus, even across populations. Like there might be, you know, certain things we should be careful with then in that case. And it kind of also shows that, you know, this view of maybe wanting to put names on it, it's really, you know, it's very intrinsic to us because we like to, you know, make statements based on that. And for instance, you see in the literature a lot of like that bacteroidetes, firmicutes ratios, which are associated with a bunch of things, apparently. But then, like often that kind of lose that strain heterogeneity or the divergence, evolutionary speaking, that has occurred there. Like, you know, those bacteria have lived in very different environments. So that's kind of something you might want to look at as well on your own. And you have also some space here to add any other analysis. You can go through the QIIME2 plugin, so maybe look at a certain particular analysis that you want to do as well. And yeah, that's it for actually the material for today. In terms of the course, we will still be having an amazing talk by Ajish Jha after that. We'll have some time for some Q&A here right now, but just to reiterate, yeah, go through, annotate that phylogenetic tree now with the taxa, the abundances, the samples, all the metadata, which will create like also some quite nice visualizations. You will be able to dig in very much in detail what's happening to particular ASVs across different communities. So that's something to look at maybe. And there's a lot to learn from that as well. And also to tease maybe a little bit what will be happening tomorrow, another question is, even if you would observe the exact same organism or the same microbe in two different, you know, the gut of two different individuals, would they actually behave the same? Because we know that microbes, you know, are very adept at reacting to changes in the environment. So like almost all bacteria have different ways to obtain carbon or nitrogen and to translate that into biomass, but that will also kind of change how they act in that environment, for instance, which metabolites are produced as a side effect of that. So one question would be, if you had exactly the same organism, but would it put it into the gut of people that live very different lifestyles or live in different communities, would the functional output or how that particular microbe acts actually be the same, or might it be pretty different? And that's something that Nick will be talking a lot tomorrow, and will show some really cool approaches and how to do that, even when you have only limited data available. So that's just a little teaser for what's going on tomorrow, and I hope that, yeah, you will have some fun with that as well. Yeah, apologies for if I went too fast with certain parts. But yeah, there's also the Slack channel. So if anything seems unclear, feel free to write it there. That Slack channel will be open for a long time, even after the course ends. So even if you see that it's in a video recording right now, feel free to ask questions there still. And I will be there as well and answering questions. And yeah, happy to receive any feedback that you have as well. And with that, I just want to also thank like all the people that participated in the course, and Sean kind of alluded to that as well. But just the fact that you just saw my face now, maybe more than you wanted to, does not mean that this, like the course material or any of the things that you did today were, you know, just designed by one person. A lot of different people were involved in creating all those teaching materials and making it accessible. And then there's also a huge amount of work that went into just making this event work, to kind of, you know, providing live streaming, setting up like the TAs here, you know, in our meeting room, organizing the events, managing registrations, and so on. So I just want to give a shout out specifically also to Dominic, who's the event and manager here at the ISB, and did a lot of, you know, work behind the scenes to make this work so smoothly, but also like a lot of the ISB team here that has worked like tirelessly to make this like work so well over the years as well. So thanks too much also like to you joining us today. And yeah, I think we have like a few minutes for some, you know, major Q&A questions if something popped up. So feel free to post them in Slack.
Yeah, so I think one that came up was this controversy about prayer action.
Yes.
Appropriate. What are your thoughts on that?
Oh yeah, that's a big topic for sure. Yeah, there's a lot of arguments to be made about it. So, and I'm not even sure if I'm the best person to make them, but yeah, one one of the things that can be said about rarefaction is that since you randomly subsample, you often generate one random sample, which can just mean that, you know, by bad luck, you might just pick out, you know, a lot of reads from a particular taxon from like a larger set of reads, and that can kind of skew your results a little bit. So like every time you run the rarefaction analysis, your results might look slightly different, and you can actually see that in the course yourself. If you just rerun the phylogenetic, like core phylogenetic steps, like the data visualization, which looks slightly different based on that subsampling. Then there is an argument to be made of, you know, leaving out data, right? If you subsample a sample with 100,000 reads to 10,000, you leave out 90% of the sample. That's just what happens. So that does not mean that what you extracted is not representative of the sample, but that question is there. So a lot of discussion is going in that. So I think it's, yeah, I think the important thing is that you're very, you know, if you write it up in your paper or something, be definitely very vocal about what you did, what you normalized, and be aware that there might be like effects of that as well that are not really a signal in the data. There's different methods to normalize as well. They make certain assumptions, which might or might not be true for your particular samples that you have. But yeah, there's a lot of literature on that as well, and we can maybe like post a few resources on Slack. I would say yes, but I wouldn't really say, I'm not sure if we're really at a point where we're completely decided in the field what it is. I do think that for like just for visualizations, like doing the PCoA's plots and so on, it might work pretty well. Like there you do see that it, you know, it kind of works, but there's also alternatives, yes. But yeah, sorry, I don't really have a real definitive answer for that.
One other thing came up is, what do you think about the new taxonomic? I think it's that's similar to
what do I think? Yeah, complicated. Yeah, okay. The question was like, what do we think about the renaming and the new naming scheme? So yeah, again, it's obviously it's hard to remember what taxa do just based on the name. So like I argued, like sometimes the name isn't also that informative anyways, right? And the end strains from a particular genus might act very different in different environments. And so it's like hard to really assign function just based on a name. Even though it makes it harder to maybe recognize things you've seen before. But I think a lot of that is also done to kind of get more consistency in how we do taxa. So kind of getting monophyletic groups. So meaning like getting really distinct naming schemes based on groups that really have one common ancestor. And as more data is coming in, those change, right? And what we first considered, for instance, one Prevotella genus might actually make sense to subdivide it into 5 different genera, because we actually see that's a lot of divergence happening. So I think it will be always the case that taxonomic naming evolves as more data comes in, because essentially those trees, like we did it in our case, are generated from genomic data. They're not really generated from just a small 16S fragment. They use a larger amount of like very conserved proteins. But yeah, I think it's something we will have to live with, that those names tend to change. But yeah, then if you really want to get around it, you can, you know, analyze everything on the strain level, and then it's all their own. Okay, I think if there's no other pressing question, we can actually take that time for a little bit of a longer break before, since we have gone through quite some material, so everybody can, you know, relax a little bit and recharge. And like I said, if you have any questions, please ask them on Slack. We'll be happy to answer. And if you're taking that course later on, then yeah, I hope you enjoyed it as well. And also feel invited to still send in questions, even that's like a week after the course has ended. And with that, we'll be going into a bit of a longer break. And I think we'll be back at around 12:30, I think. 12:45. Okay, yeah, 12:45 we'll be back. So that will be in around 37 minutes. So yeah, thanks so much for joining us. I hope it was useful to you. And yeah, let us know what you think. Hello, everybody, and welcome back. So it's a huge pleasure and privilege right now for me to introduce our first speaker in this series. And while I do that, Dr. Jha, please feel free to unmute yourself and start sharing your slides as well. So Dr. Ajish Jha is an assistant professor of biology at New York University in Abu Dhabi, where he leads the genetic heritage group. Dr. Ajish grew up in Kathmandu, Nepal. He received his bachelor's in molecular and cellular biology from the University of California, Berkeley in 2007, his PhD in human genetics from the University of Chicago in 2015, and completed his postdoctoral research at Stanford University in 2019. Currently, his research focuses on integrating population genetics and microbiomes of traditional populations in the United Arab Emirates, Nepal, India, and Oceania. His work has spanned the microbiome, genomics, bioinformatics, immunology, evolutionary biology, and cancer biology, which has resulted in multiple peer-reviewed papers. He has received funding from NUAD, Stanford University, as well as the Bill and Melinda Gates Foundation. So thanks so much for presenting today. And yeah, please take it away, Dr. Jha. Dr. Jha, I think you're still muted.
Sorry about that. Let me share my screen. Can you see my screen now?
Yes, looks good.
Okay, thank you so much for a great introduction, and thank you for the opportunity to come and talk today. It's super late here. It's actually midnight, so I may be a little bit rusty in my Zoom skills. But today I'm going to talk about scaling up multi-omic studies to identify functional contributions of microbiomes on health of underrepresented populations. And those of you who are here in the audience, you probably know that this is super important. That's probably why you're here. So the last few decades of research in human microbiome has been very productive. And what we now know is that gut microbiome varies significantly across lifestyle, right? So in this paper published in 2017, this group of researchers, they looked at gut microbiome variations across a lifestyle gradient. Each dot here is an individual. They're color-coded by the populations they come from. Lighter colors are populations from or that are practicing traditional lifestyle. Darker colors are populations from industrialized nations. And as we can see, there's a stark differences in gut microbiota across these populations. Recently, we published a paper in which we looked at gut microbiome variation in children. And what we found was that initially the gut microbiota of children across different lifestyles doesn't vary much. But by 3 years of life, the gut microbiome is starkly different. So in this right-hand side plot, the circles, the red circles indicate individuals coming from non-industrialized populations like the Hadza. The blue, light blue color dots indicate populations coming from transitional populations, and the blue, dark blue dots on the top, they represent children from industrialized populations, right? So there is a massive variation in gut microbiome across lifestyles. Oops. What is going on? Okay. But what we also know is gut microbiome development is an ecological process. So in this particular study conducted in Italians, what the researchers found was initially, if you look at the circle in the middle, the pie, initially in a child, a one-day-old child, gut is colonized by different types of bacteria that come from a variety of sources. The red outer circle indicates bacteria in the child's gut that look like an adult gut microbiota. By 4 months' time, though, more than half or about half of the child's gut microbiota resembles the microbes in an adult gut, right? So what this means is initially Microbes are like, hey, look, it's an open space, let's go make home. And they are stochastically, they're just, every microbe is trying to colonize the gut. But many of the microbes are not very successful in this colonization process. So by 4 months of time, the successful microbes are the ones that are traditionally known to live in the gut, right? So this process is very important. By 3 years of time, the child's gut microbiota starts to resemble an adult microbe. What we know is that the adult microbes interact with many human physiological processes. So in this figure on the top in the green section is gut lumen. That's where all the microbes live. In the middle, the cells with finger-like structures, these are epithelial cells in the gut. Those cells prevent microbes from getting into our bloodstream. Although the microbes cannot get into our bloodstream, they produce a variety of different chemicals that can get into our bloodstream, and these chemicals can go and influence a variety of different physiological processes in our brain, in our organs, and whatnot. So the imbalance in any gut ecosystem can lead to disease. So there's a paper that came out from Sean's group, actually, the Monroe et al. Paper. They looked at association between gut microbiota, individual taxa, and a variety of different human phenotypic traits. Some of them related to disease on the top, shown by orange box. Some of them are related to a healthy lifestyle on the bottom. So red means, so in each column is a bacteria, each row is a trait. Red means the bacterial abundance is elevated in these traits or is positively associated with those traits. Blue means they are negatively associated. So we see a whole bunch of bacteria that are associated with disease and healthy traits, and they are sort of opposite to one another, right? So when they are positively associated with health, they are negatively associated with disease-like phenotypes. This indicates that gut microbiota are strongly associated with markers of human diseases. Gut microbiomes are also associated with treatment efficacy. In the right-hand side plot, Zimmerman et al., they looked at 76 gut bacteria isolated from the human gut, and they asked, can these microbes metabolize drugs? So after 12 hours, they found, so black means 0% of the drug was metabolized, white means 100% of the drug was metabolized. If you look at the top and the bottom of this heat map, you can see that there are a lot of white regions, right? So some drugs are metabolized by all of the bacteria on the bottom. On the top, some of the drugs are metabolized by some of the bacteria, and a lot of the drugs are not metabolized at all. So what this means is, given your microbiome composition, gut microbiome composition, the treatment efficacy in a diseased individual may vary. So this type of research has led scientists to sort of come up with sort of like a personalized nutrition. So in this particular study, Zivi et al., they looked at or they measured a variety of different features in 800 participants. One of the features was microbiome, and by putting all these features together, they were actually able to design personalized diet to lower glycemic responses. There are actually startup companies out there today that use either your genetic data or your microbiome data to design personalized nutrition. And these companies are expected to have a massive impact in healthcare. So this is great, but all of these, ecological succession, association with disease, personalized diet, is only relevant currently to people of European descent. Why is that? Because most on the top plot here, in this paper published earlier this year, what this group of scientists found was that most of the samples on which microbiome has been characterized come from European individuals or individuals of European descent, right? Even though Europeans constitute a very small fraction of the entire world population, most of the samples come from Europeans. This is improving at the bottom. They looked at what proportion of samples come from different world population over time. 2010 and 11, 100% of samples came from Europeans. In 2020, there is a little bit of diversity there, but still dominated by Europeans. But I think the problem goes beyond the number of sample or sample proportion. I think there's a problem in how we sort of approach microbiome studies. So the first step in microbiome study is characterizing the microbial variation. Second step is to then say, okay, if there's some variation exists, is this variation associated with some trait, like the Monod et al. Paper? The third step is, okay, there are a bunch of microbes that are different. We could predict their function, but to make sure that these predictions are true, we need to layer additional omics data sets. So these omics data set may be metabolomics, metatranscriptomics, metaproteomics, whatnot, right? So then we understand by layering all these different omics data sets, we then understand what microbes are there or not there and what they're doing or not doing. And then that sort of leads to this translational research that can help the human society at large. In the case of European microbiome studies, all these are happening and translational studies are being done at a very rapid pace. But in traditional microbiome studies, we stop at characterization. The number of samples are increasing, but they stop at characterization. There's not a lot of studies looking at association between microbiome and traits, microbiome and disease, not a lot of studies looking at functional data, and very few translational studies. So then the question is, why is that? One answer could be the prevalence of chronic diseases are very low in traditional populations. Second scenario could be that chronic diseases exist, but they are not associated with microbiomes in traditional populations. The third option or third scenario is that both of these are wrong, but there are so many barriers to conducting this sort of functional or translational studies in traditional populations that we never get there. So last few years of my research has been focused on trying to figure out which of these scenarios are true. So what we have done is we've visited a number of traditional populations in Nepal, which is the Himalayan country. So on the top left is a photo of one of these traditional villages. We've sat down with the community leaders. We've discussed with them the scope of our study, the scope of microbiome study, the importance of it for participating populations. We've collected a lot of survey data, sort of characterizing people's diet, use of medicine, and whatnot. And we've also measured some phenotypes. And the analysis of this data shows that chronic diseases are prevalent in traditional populations. So in this plot, the histogram plot on the left, what we found was systolic and diastolic blood pressure both are highly prevalent in the indigenous populations of Nepal, who are severely underrepresented in microbiome, genomics, both literature. BMI, or sort of like high or overweightedness or obesity is not that high, but hypertension is very high. And if we look at neighboring country, India, what we find is that hypertension is massively prevalent. In fact, cardiovascular disease is the #1 non-communicable disease that causes deaths in India. High blood pressure or hypertension is the primary risk factor for CVD, and hypertension is highly prevalent among the poor and marginalized populations. So on the right-hand side here in the map is a sort of representation of prevalence of hypertension in India. Red means 10 to 15% hypertension, right? So almost all of India is red. On the left-hand side or right-hand side within this green circle is the state of Assam, where hypertension is prevalent at 15 to 25%. So we collected samples in Assam, where our collaborators in India did this study. They collected the samples, they generated 16S data, and I helped them analyze this data. And in a paper that was published earlier this year, we found a robust association between systolic blood pressure and microbiome composition, indicated by PCoA on this plot on the left, right? So what this means is chronic diseases are prevalent, and at least hypertension is associated with microbiome, even in traditional population of South Asia. So the third scenario seems to be the true scenario where there are many barriers to conducting microbiome studies in traditional populations. And what are these barriers? These barriers exist in every step from sample collection to publication. So there's very limited funding, or in many cases, complete lack of funding to look at microbiome of traditional population, in-country funding. Most of the funding in country or from international agencies focus on infectious diseases, and there's very limited understanding of microbiome and its effect on human health within the funding agency, regulatory agencies, and sometimes among the scientists in the low and middle income countries. It's also very difficult to collect samples because there is lack of health infrastructure. So there are not a lot of hospitals, and people live in remote geographic locations. So people don't usually come to hospitals until it's like an emergency situation, right? So collecting large number of samples is very, very difficult. And who will collect the samples? We need trained people to collect the samples, and it's very difficult to find trained field staff. And even if you go out there to train the field staff, there's a high turnover, and it's extremely difficult to sustain a trained workforce from personal experience. Whatever number of samples you collect, when you bring them to a university or a research institution to do lab work or genomics research, we have to face the scenario where there is very limited research infrastructure. We have very few trained scientists, and even if you train scientists, they also leave. Sometimes they leave to do PhD abroad. Sometimes they go and find another type of job. So it's very, very difficult to train, sustain a trained workforce. Bioinformatics analysis, the capacity to do that is virtually non-existent in many low and middle income countries. There's very limited computational infrastructure. There's not a lot of training provided at master's or undergraduate levels. So hard to find experts or trainees who are interested. And even if you train some people, again, they also leave for better jobs abroad. So all this culminates into limited number of samples that we can get, and one-dimensional data is what I call it. So basically people may do 16S or maybe some metagenomics, but just that, right? We cannot have, there's not a lot of opportunities to generate multi-omics data, and which equates to limited novelty. And limited novelty equates to less chances of publishing in high-profile journals, which results in limited funding for subsequent studies, and we go back to the top again, and this cycle repeats. So this happens at a very reasonable frequency when we start thinking about traditional and underrepresented populations. So what we have done in the past, since 2019, is we've tried to develop this systematic approach in scaling up functional studies in microbiome in underrepresented population. So we published a paper in 2018 in which we compared gut microbiome variation going from foraging populations on the left in this heat map to farming and industrialized populations. And we see massive shifts in gut microbiome. So farmers living in Nepal, their microbiomes are starting to resemble the industrialized populations, right? So there is this massive shift. So using this as a foundation, we have been successful in obtaining a grant from Bill and Melinda Gates Foundation, which allows us to go all the way to the functional level. So we're asking 3 questions. So we're conducting 2 studies looking at the functions of gut microbiome in underrepresented population. One is in Himalaya, one is in the Emirati population here in UAE. And the first question in the Himalayan study we're trying to ask is, how does the gut microbiome develop in infants across lifestyle? Because like I said earlier, the ecological progression is very well studied in Europeans, but we don't know how the progression happens in underrepresented traditional populations. So this is sort of like characterizing the gut microbiota. The next step is to look at association. So in Nepal, 33% kids actually end up stunted. It's because of undernourishment, other factors, right? So Gut microbiota are causally linked with stunting in other populations. So we want to ask, are there associations between gut microbiomes and stunting in Nepali children? And if there is, the third step is what is the biological mechanism that leads to gut microbiome dysfunction in stunting? So to answer these questions, we're collecting a massive trial in Nepal, or it's not really trial, it's a observational study where we're collecting samples from pregnancy, first trimester in pregnancy, it's a longitudinal study, to the end of first year of life in kids. So we're collecting maternal lifestyle and diet using survey questionnaire. We're collecting maternal gut or fecal samples for gut microbiome analysis, vaginal microbiome, and we're also collecting using survey questionnaires if various factors that influence the child's life in the first year. So for example, child's birth mode, what this kid is eating, breast milk, formula, non-formula food, medicine, vaccination, and other sort of environmental factors. So the goal here is we want to use the metagenomic data or 16S data to understand how gut microbiome progresses. Maybe it's different in different lifestyle, and maybe this sort of the dysfunction will lead to some association between stunting and particular microbes. Once we figure out which microbes are associated with stunting or low birth weight, we can then extract those microbes because we're collecting all of these samples using liquid nitrogen, so they're flash frozen. So we can isolate these strains and do functional characterizations in the lab. So here is our sampling scheme. We're collecting samples from 4 populations: foraging population, a farming population, a peri-urban population, and an urban population. On the top is actually the sampling scheme. So we're collecting samples from controls. Basically, these are women of reproductive age who are non-pregnant. So we're collecting samples in the 3 samples every 3 months, 1 sample every 3 months. And we're collecting samples from pregnant women at time point 1, time point 2, time point 3. Red dots here means fecal sample. Saliva, we're also collecting it. I'm not going to get into it. At birth, we're collecting vaginal swabs. And then for the first year of the child's life, we're collecting breast milk at 1 month interval. And then we're also collecting gut microbiome or fecal samples from infants. In the first 3 months, we're collecting samples at every 2-month interval. And after that, we're collecting samples at 1 month interval up to the 12th month. Despite COVID, we have completed sampling from the first trimester to the 6 months of the kid's life. We're collecting currently samples from 7th to 12th month, and we're hopeful that we'll get funding to continue this to the first 3 years of life. To sort of give you, oh my God, this is covered here, but to give you an idea of the scale of this work, we are collecting 50 mother-infant duos from each group. We have 4 groups, and there are 21 time points, which results to about 4,200 fecal samples that are flash frozen. We have also 200 vaginal samples, and we have about 2,400 breast milk samples. Collectively, this results to 6,800 biological specimens, and we're collecting samples in multiple replicate tubes, which results to, in our estimate, about 19,000 tubes of samples from these 4 underrepresented populations, which is a pretty massive number of samples to collect using flash frozen medium. So we are currently this is an ongoing study, it's not completed yet, but once this is completed, we're really hopeful that this will result in meaningful discoveries that will be useful in addressing stunting problem in Nepal. The other study that we're doing here is looking at the oral microbiome as a part of the UAE Healthy Future study, which was funded with a local investment funded by the UAE government. So for this study, we have collected DNA from, we collected spit samples from, mouthwash sample from 27 individuals from which we extracted the DNA. The optimization to extract DNA from mouthwash samples took us a little bit of time, but we've done it. We're currently doing 16S and metagenomics sequencing from these individuals. And we also have 300 clinical traits, right? So these are blood markers, anthropomorphic traits, other sort of disease-related markers from each of these 2,700 individuals. We're in the process of also developing metabolomics. I put a black box here because I have no idea how to work with the metabolomic data, but it is important. So we're sort of inching towards learning how to integrate metabolomics with the metagenomics data. So at the end of this study, the goal here is to identify link between oral microbiome and disease and understand the biological mechanism by which oral microbiome may influence the disease. So with that, the summary of our work in the last 3 years is that we know that human microbiome as an ecological process is very important, and we need to understand it in detail to sort of understand how microbiome and human health are connected. We need functional studies linking human microbiome to diseases in traditional populations because those types of studies are scarce. But there is urgent need to start these studies because these types of studies benefit the participating populations, right? So NCDs are rising rapidly in non-industrialized countries, and without these types of studies, there is a chance that that these countries, their health infrastructure may collapse if we cannot prevent NCDs from rising. Despite all these obstacles, I think these two studies show that it is possible to conduct large-scale functional microbiome studies that benefit these underrepresented populations. And this is especially possible if local stakeholders are involved, like if the government regulatory agencies are involved or funding agencies, local funding agencies are involved. With that, I want to thank, I have a lot of people to thank. I'm not going to go into each person, but there are a lot of people who helped me do the field work, are currently helping me. There are people involved in laboratory work. I definitely want to thank Biswas Chepang, the autocorrect messed it up. His name is Biswas Chepang, and the female community health volunteers in Nepal who have been massively helpful in recruiting participants. A lot of collaborators here, but I definitely want to acknowledge Justin Sonnenberg at Stanford, with whom we're running these some of these studies together. Funding sources, I cannot thank the Gates Foundation enough. And we are very, very, very thankful to the participants for welcoming us to the communities, as well as the Nepal Health Research Council, who gave us permits to conduct these studies, Stanford and NYU IRBs as well. So with that, I want to thank you for your attention. My lab is called Genetic Heritage Group. You can reach me by email or through Twitter. If you have any questions, we're open to collaborations. And if there's any recently graduated PhD students and looking for a postdoc, we're also looking for a few postdocs. So with that, I'll take questions. Thank you.
Hi. Thanks so much, Dr. Jha. That was a great talk. We have a little bit time for question, but probably don't want to take up too much of your time either to allow you your like well-deserved rest. There was one question I saw in the chat that was asked by Amber-Ree, who was asking what is being referred to when we say traditional microbiome studies versus European microbiome studies. Are we just referring to non-European population?
Yeah, so the difference there is when we say, well, so sort of like European is synonymously used to indicate like industrialized population, right? Because most of Europe is pretty industrialized. So when we say traditional population, we're talking about populations that live in non-urban areas.
Awesome, thanks so much. Yeah, and yeah, I was also interested, you know, about your experiences in terms of community engagement, like so what did you think, for instance, like was particularly important there, but also like what additional information you might have gotten from engaging with community representatives and building up those collaborations? And yeah, that would be my question.
Sure, yes. So community engagement is super important, but how you define community engagement may differ, right? So in our study, those ongoing studies in Nepal, what we've done is we've hired people like our sample collectors are people from within the community. We have gone through extensive length to actually identify, nurses from that community, and we've hired them so that people feel comfortable, participating in our studies, right? And that not only from our perspective, but when you hire people from the community, they are willing to go an additional mile to help their own community. So that is super important, right? So in the past, what we did was we sat down with people, we talked to the community members, and we collected samples, but I did the sampling myself. So there was community involvement, but they were not involved, they were not actively engaged, right? Right now, we're hiring people year-round to go and collect samples for us. We've trained them, and they're from that community, so they are stably there. And they're actually making massive differences that I had never thought about. So and I know we're short on time, but I think this is important, so I'll discuss this. So we were thinking about malnutrition, gut microbiome, identifying causal bacteria and all that, right? But one of the things that we did not realize was we can give back to the community in real time. So one of the things that we can give back to the community is Nepal government actually has a program. It's a government-related program. So all mothers who do 3 prenatal checkups and do an institutionalized delivery, so not delivering the baby at home, but in a hospital or a healthcare facility. If a woman completes all those 4 milestones, she actually gets like a few $1,000s from the government, right? And this money is not that important, but what is more important is it actually influences the child's health and the maternal mortality, both, right? Maternal and child improves those 2 things. So these women who we have hired, all of our sample field workers are women. They actually are checking and making sure that these women, our participants, are going to are sort of like fulfilling these milestones. And as a result, we have like about 80% of our participants have actually fulfilled this milestone. And overall average is like around 30% or 40%, something like that, right? So that is not something that we had thought of before we started this. This just came organically from our team who's working in Nepal, and just like, it's a massive contribution to the participating community, which I'm very, very proud of.
Yeah, thanks so much. Yeah, maybe we have time for one more question, and then you'll be off to be able to sleep finally. But there was a question by Noura El Habal, who was asking about the infant microbiome study. So she was asking if you have data on maternal diet or medication, and then also about the whether is also measured during lactation. I might have missed this, but during which times do they collect milk samples, and do they take into account infant feeding?
Yes, so we do have data on the mothers. So we actually have 4 different survey questionnaires. Each survey questionnaire has 150 to 300 questions. It's massive, I know. Our program officers, they complain about it, but the point of using the survey questionnaire is there is no data from these populations, right? Like nothing. Like we don't know what they do during pregnancy. Is there a special diet that they use? What happens in the child's first year of life, how much interaction they get with like animals, with other. So basically we have 1 survey data to look at the pregnant women. And then we have another survey looking at the child in the first 3 months. And then a third survey data from 3 to 6 months. And then a 4th survey questionnaire to capture lifestyle related covariates from 6 to 12 months, right? So in using this survey questionnaire, we can get a good sense of what the mothers and infants are eating, whether they were vaccinated or not, whether they were medicated or not, and so on. So what time the breast milk sample is being collected? All of the samples are being collected in the morning, because people live in very remote areas and we don't have the luxury to just keep visiting the same person multiple times during the day. So our field officers, they go to the participant's home before 6 a.m. So the participant produces the fecal samples, and then, she just hands the fecal samples to our field officers. The fecal samples are dropped in a liquid nitrogen tank. We have a small portable liquid nitrogen tank that that people carry door to door. And then the breast milk is also collected at that time. So we don't have an estimate of whether, it was a recently fed milk or not. But yeah, that's one of the limitations.
Yeah, perfect. Yeah, and with that, let me thank you again for taking the time to speak to us today. It was like a huge privilege and an amazing talk. And a huge virtual applause to Dr. Jha from our participants here. And thanks for joining us today, and have a great night.
Thank you so much. Take care.
Yeah, and then I think we'll go right ahead to the closing remarks by Dr. Sean Gibbons again. So stay tuned for that right now.
Thanks, Christian. Well, that's a wrap for day one of the course. The third year we've done this. Thanks so much to our sponsors, Illumina, Metabolon, and the Washington Research Foundation for their support. And, a huge thank you today, I'm sure all of you are very thankful as well, to Dr. Christian Diener for walking us through the course and teaching us a ton today. And to our amazing speaker, Dr. Jha, who really showed us, the challenges associated with doing this kind of research around the planet and different populations, and examples of how it can be done well and how it's being done currently. To recap, we introduced you today to some computational tools for Amplicon sequencing data analysis. In particular, we introduced you to QIIME, this whole software ecosystem, which includes functions for processing, analyzing, and visualizing data. We focused on microbiome data from 3 indigenous populations from across the globe. If you're stuck with any of these materials, please reach out using the course Slack channel, and the TAs will jump in and help you out, even after this course finishes up right now. And if you wake up on the other side of the world and watch this on our YouTube stream, we'll try to get to your questions when we're not sleeping, essentially, as soon as we can. Feel free to share your experiences using the hashtag #ISBmicro22 on Twitter or Facebook or whatever you social media on. You can tag ISB on Twitter at ISBSCI or ISBSci. We look forward to hosting you here again tomorrow for the second day of the course, taught by the very talented Nick Bowman. We will leverage the processed data from today's course to explore how dietary variation can have a huge impact on the functional outputs of the microbiota. So we'll kick things off at 9 a.m. Seattle time or Pacific time zone tomorrow morning. We hope to see you there. If you want to learn more about us and about our institute, you can go to isbscience, all one word, .org. And with that, thank you so much for joining us, and have a great rest of your day.